Mohanta sir. Good morning, sir.
How are you?
Fine, sir. Fine, sir.
Good, good.
Yeah, sir. Principal may not be joining right now. 9:30, 9:40.
Okay, okay.
So he told that he'll be joining some more late, sir. 9:40.
This
Sir, diploma pharmacy inspection 2 days.
from
Maybe government officer.
Andhra Pradesh.
Yeah. Yeah.
Our Ramalingam sir.
Ramalingam is also there along with principal, sir.
Oh, okay, okay, okay.
Sir, if possible,
Pardon?
make it, sir.
I didn't get you.
Sir, the virtual background, sir.
So what I have to do?
Sir, you have to download it, and in the Google Meet icon, there is 3 dots are there. So click it.
1 minute. 1 minute.
Yeah, yeah.
Downloaded.
Yes, sir. The icon of screen sharing, there. Yes, sir. There.
3 dots.
Yes, sir. There, change background will be there. You have
Whiteboard.
to change the background. You have to click it.
How to locate that? It will be there in my download.
Yeah.
It has come as a picture. Yes, this picture has come. Huh?
Yes, sir.
Uh, slightly blur your background.
A symbol will be there, sir. You can add.
Yes, locate the plus symbol.
Yes, sir. Sir. Yes, sir. Right, sir.
Yes, now background
visible,
is showing.
Clear, sir.
You are able to see, no?
Yes, sir. It
Background.
is clear. Clear, sir. Clear, sir.
Yes.
Sahiti?
So,
2 minutes, sir. 2 minutes. Saiti, whether we can
Good
go
morning, sir.
for
Good morning.
good morning, good morning. Participant reminder. So,
So, good morning.
you can go for participant reminder, followed
Yes,
by
sir.
Mohandas sir introduction.
Yes, sir.
Yeah.
So, good morning to the respected speaker and the fellow participants. A warm welcome to the sponsored online faculty development program for effective monitoring and transition of care. Before going for the session, I just want to remind you that many of the people are not entering the registration link. So already the participants are provided. So I'm very honored to introduce our speaker for today's session, India,
Breaking the
former
voice.
officer.
Saiti,
Yes, sir. Yes, sir.
it is unclear from your side. We can start once again.
Okay, sir.
Yeah, now it is clear.
Okay, sir.
Yeah.
So former National Technical Officer, WHO Country Office for India. Guruprasad Mohanta having 33 years of experience in teaching and research, and in his professional journey, Sir held various academic positions such as member of National Level Advisory Committee, Government of India, member of Board of Studies for various institutions, and for expert member, Administrative and Social Planning and Ethics Group of Federation of Asian Pharmaceutics Association. And Sir was a technical officer, essential drugs and medicine in the projects funded by the EPN Health Action International attended various training programs in WHO and FIP, and Sir had been principal author and co-author for 14 books, and has published 128 papers in international journals and 80 papers a personal New Indian Express for 4 years. And Sir has also given a radio talk on ORG his contributions, namely recipient of AIPST Pharmaceutical Award 2011-12, and has received various grants and UGC fellowship. And invited as expert for various committees and conferences. With this brief never-ending knowledge to all the participants with his profound experience, Sir is a person who is actively involved in engaging the community health. So to be inviting Guru Prasad Mohanta sir.
Shall we start?
Yes, sir.
On that? Or
Sir,
we'll
we
wait?
can
We
start.
can
We
start,
can
sir.
start.
We can. So good morning, friends. Thank you, Dr. Mohan Raj, Chaiti, and all others. And I take this privilege to thank the organizers, especially the principal sir and the Ramalingam sir for giving me an opportunity to share I just looking back to the period when I was with World Health Organization. I how to introduce the pharmacovigilance program in this country at national level. And that was the period when the World Bank capacity building project was just ended. So the country did not have. And it controller of India, who was my counterpart from the government of India side or so one day I just implementation of the program, and one day I told, sir, we have, and it is now for us to go ahead to have our own program. He used to tell Mahanta, don't worry, we'll have. And one day he said, and when I explained him this is how we need to proceed, he immediately spoke to the then Joint Secretary that we want to introduce as a country program, not as a sub. And the Joint Secretary immediately said, go ahead. And to begin with, what he said is, let us introduce is as a mandatory to medical through changing the requirement of Medical Council of India. Made is pharmacovigilance activity in every medical college as a mandatory requirement to get MCI approval. And that was the beginning of this of this country that is currently being implemented. And fortunately, instrumental in implementing these programs, introducing these programs, do not find them in any of their contribution. Yes, personal interest to introduce this. And that is how we see the pharmacovigilance program at this level, where we are recognized as a center of World Health Organization. Had at least I like to acknowledge their contribution. One is Dr. Surinder Singh, the then drug controller of India, and the then IPC General Secretary. And that is how we see. Because many of our young teachers may not know about the genesis of introduction of the National Pharmacovigilance Program. Yes, friends. Tell you, I have not studied the Raj has done. I have just studied pharmaceutics, and it is my passion It is my love, my interest that make me to look pharmacy from a different angle, and I serve this country as an officer on essential medicines. And I do not know in World Health Organization in other countries, World Health Organization in the Geneva office, there are a lot of pharmacy people are working. And in fact, the head of this, the safety wing, is one of our pharmacy teachers from India. Dr. Santipal is the chief of safety monitoring program. But in India, we do not consider pharmacy as a part of all these issues for many regions, and it took time even for me to make others understand that the pharmacy also deals with the public health. Pharmacists are also concerned with the health of the people. They are not just the traders. And one person asked me a question when I joined WHO. One of my colleagues asked me, you are a pharmacist, and pharmacists are involved either making medicine or selling medicine. Either making medicine or selling medicine. And WHO does not do any of this. So what are you going to do in WHO? One of my friendly colleagues. So it took some time for me to make him understand what public look at pharmacy, either selling medicine or making medicine. Pharmacy is much beyond that. My screen is visible?
Yes, sir, it is visible, sir.
Okay,
It is
friends.
visible.
So I begin with my presentation with two quotes. One from the Napoleon Bonaparte, who said, I do not want two diseases. One is nature made and the other is doctor made. The great philosopher Oliver Holmes said, I firmly believe that if the whole of the materia medica as now used would be sunk to the bottom of the sea, it would be all better for the mankind and all the worse for them. So these are all people think medicine are not safe. But friends, in spite of all these criticism about the medicine's adverse effect and side effect, now the longevity has increased to that extent. We are able to fight even the COVID just because of medicine. Yes, we can improve the medicine so that we can minimize the risk involved with medicine. Risk will remain always, but definitely we can minimize it. Friends, my presentation today consists of these 5 areas. Why teaching pharmacovigilance is a concern. What to teach, how to teach, the concept of teaching to learning, and use of artificial intelligence in pharmacovigilance, preparing for the future. These 5 areas I will be talking. Friends, why we are concerned, why we need to talk about teaching pharmacovigilance when we are not talking much about teaching pharmaceutics or teaching any other subject, but why pharmacovigilance? Because it is a new subject, relatively new subject. You know, for the first time, the terminology pharmacovigilance was introduced in France in 1960s. and to get a wider introduction of this pharmacovigilance word, it is only in 2002 when WHO has introduced. And from then onwards, the pharmacovigilance, the subject got some weight, some interest in health science. Many of these teachers, especially those who have completed their programs prior to 2008, who are now teachers, they never studied. Friends, when I studied, I never heard the word pharmacovigilance. Of course, I was a student of pharmaceutics, but the subject was not familiar with us. Name of the subject itself was not familiar. Leave about the contents of it. So that is why we need to train the present generation teacher on pharmacovigilance. Second, for every job we do, they give an training. Whether we go to the industry, we go to the regulatory side, everywhere they give some training. So prior training is important, but it is the higher education, like what we are, directly we are coming from the institution where we have studied to become the professor in a college or in the university. And during the student time, we have very little understanding of the education system or the teaching learning process. But we are directly made professor from the education to the institution. And we know how difficult it is. Friends, people may think teaching is one of the simplest job, but I do believe, and I'm sure you also might have experienced, it is not simple job because we are dealing with people. We are dealing with people, and who are the future? So what to teach? Yes, we are guided by the curriculum. We have a syllabus. We are guided by that, and the syllabus is to be taught. But friends, you look at the pharmacovigilance, there are many institutions which offer certificate program, to be part of either value-added courses. Now the new concept has come that institution must offer some value-added courses. Yes, syllabus is there, but in addition to the syllabus, one must have the value-added courses. So many of the institutions have started the health science institutions like medical science, dental science, nursing science, and of course our pharmaceutical science. So it begins with a certificate program, two part of a unit, one chapter in a to an elective course. If you see now BPharm program, one of the elective course is pharmacovigilance. So that means we have a variety of programs of different level. So always we have confusion how much we should teach. Friends, you all know there is a WHO collaborating center, that is Uppsala Drug Monitoring Center, but there is a center for pharmacovigilance, education, and patient reporting. And this center is at Lareb in Netherlands. And this center is the collaborating center for education, pharmacovigilance education. I was fortunate to be part of some expert meeting programs how to teach pharmacovigilance in various health science program. And this is how I participated. This was planning, educating the educators, how we can teach our teachers. And a couple of years back, I happened to be one of the invitees to participate along with Dr. Kalai Selvan, who will be addressing you probably some other day, who is the principal scientific officer at Indian Pharmacopoeia Commission. We both attended from India. So this LAREB laboratory, we have this collaborating center, and not having proper training on these areas. The importance of adverse drug reactions, the risk driving areas of important medicines, individual capacity report, pharmacovigilance in clinical trial, counterfeiting, quality aspects and medication error. Yes, friends, this counterfeiting is a very important area, but we do differ with the rest of the world because we have not yet completely defined the counterfeiting in our legislations. Spontaneous reporting system, signal detection and management, post-authorization observational studies, benefit risk assessment, pharmacovigilance and risk management system, industry and the regulatory authorities, the mandatory procedure what the industry should follow, pharmacovigilance organization and public health, how we are all interested in public health, and we teach this in pharmacovigilance subjects. And another very important area is communication and the source of information. We talk communication and all this communication, and we do not teach. Unfortunately, we do not teach this communication. And communication is very, very important for us in pharmacovigilance. And the source of information. So this should be taught. This is what the curriculum designed. Of course, depending upon our job responsibility, we need to prioritize because we have limited options in terms of time available for teaching. Friends, the WHO has defined pharmacovigilance as a science of collecting, monitoring, researching, assessing, and evaluating information. So this, where from we get all this data? Data from the healthcare providers as well as from the patients on the adverse effects of medicine, biological products, herbals, and traditional medicines, with a view to identify the information about the potential new hazards and to prevent the harm to the patient. Friends, we have transformed ourselves. We have changed our focus, myopic vision, like only the medicine safety. No, we need patient safety. Medicine cannot be looked in isolation. Medicine must be looked with the patient who are the ultimate beneficiary of the medicine use. So we have shifted our attention from medication safety to patient safety, I'll touch upon this. Why do we need pharmacovigilance? First, we have, all these that in the one slide I have shown what is the scope of my presentation, and there one of the points we had is why do we need pharmacovigilance? These are all the science aspects, not the aspects of economic aspects.
Sir, one small request,
Yes,
sir.
please.
Sir, voice is not audible, sir. You can increase your volume, sir. They say the
Okay,
voice
and
is somewhat low.
now voice is okay.
Yes, sir. Okay, sir. Okay.
And now just see.
Yes, sir. Voice is okay, sir. They're asking to speak somewhat louder, sir.
Okay, sure. So I'll
Yeah.
speak louder. I'll
Yeah,
speak.
sir. Thank you, sir. Thank you.
Usually my voice is always louder, but anyway, I'll increase it.
Okay, sir. Okay.
So in clinical trial, very few samples are used from healthy volunteers to the patient. But when we use medicine in practice, there are a large number of people who are using. So it is possible to get all the information on medicine use if we try to monitor things in a greater perspective when the medicines are in use. Clinical trial, it is a control study, everything is controlled, and sometimes even the patient is also in our control. So it is a control study, insufficient safety data because the medicines are not used in all the people of the society, all sections of the society, but only on selective population. See here, at this moment, we have the problem of COVID-19, but we are facing problem because we have no vaccines for children. Because when we did the clinical trial, the children were not included, the very vulnerable population. So similarly, when we do study, usually the vulnerable populations were not included. So insufficient safety data on special population. Fortunately, we expect some study has been done and COVID-19 vaccine will be available sooner than our expectation for use by children. Broader aspect of product safety, we do not study overdosing, abuse potential, medication error, because this was not our target. Our target is different during clinical trial. Improved characterization of adverse effects, the genetic variation, enzyme stimulation, all these things are not possible to do with a limited time. While a company or an organization does the clinical trial. Friends, we have seen these medicines were introduced. Now, of course, different the bedaquiline. So these medicines are introduced even before completion of the clinical trial. And we have seen now the vaccines that we are using at this moment, they are all emergency use authorization. Complete clinical trial is not yet over. So that is why the people are more careful. The health system managers are more careful. And you might have seen when this COVID vaccination is being done, everybody is advised, wait there in the center for 30 minutes after the injections. Don't go away. This is just regarding the adverse events following immunization. Friends, initially it was only the adverse events. it was the adverse drug reactions, but now it is not just restricted to the adverse drug reaction. And we need to tell our students, see, students are always interested whether this is going to give me a job. I study this, yes, subject is very important. Contribution to the health science, it contributes. But whether it will give us a job, that is the priority in most of the students' mind. So we need to first impress them on pharmacovigilance, see it has got a wider scope. And while we take class any subject, one should be first think that we as a teacher should impress about our topic, our subjects to the students, and that is possible if we talk about the scope of the subject. Look at this, friends. The current situation, this I have collected from different sources, and this current situation, what is happening in pharmacovigilance industry in our country, not in other countries. One may start the career at a trainee junior data analyst and can go up to the vice president, director, and so on, just within a span of 10 years, just within a span of 10 years. And you can see with these 10 years, there is a substantial increase in our wages. Right? This is always a concern for our students. When we study pharmacy, another person studied engineering. Engineering graduates are better paid than the pharmacists. Very often these are all our concern. Students raise these issues with their teachers. We need to reassure our students that this is health science, and health science industries do not grow like other product industry. Our services are always regulated. Our service charges are always capped. The price of medicine, it is not that a company can fix. We have the regulatory mechanism. Government fixes. Even the drugs which are not under the control of drug price control order, the government can fix the price based on the need of the society. So the growth of the pharmaceutical and related industry is slower compared to other industries, and that is why our salary is slightly different. Friends, so this is important for us to tell where are the opportunities. Yes, even the health system, their pharmacovigilance is important, because now the NABH accreditation, government, lot of regulatory changes have come. The decision making on medicines, restricted use, something like that, or changing the indications, issuing guidelines, and all this we have at the government sector. And in the private industries, now every pharmaceutical industry has to have the mandatory pharmacovigilance activities, and in this pharmacovigilance activity, an industry has to submit PSU1, periodic safety update report. And you have seen many software companies, including TCS, Accenture, Infosys, Cognizant, they have started the pharmacovigilance activity. There are international agencies who are concerned with pharmacovigilance, and there are scope to work with these international agencies also, like World Health Organization, Management Science of Health, the Uppsala Monitoring Center. Friends, whenever we teach, we should give a historical touch, because historical touch will bring the interest of the students. And here two important incidents, international incidents, I'm trying to look at. One is that happened in 1937 in the United States of America, sulfanilamide elixir issue. Friends, sulfanilamide is a sulfur drug, and during these 30s to 40s, the sulfur drugs were the wonder drugs. Any infection, we don't nothing, okay, some wounds are not healing, just powder the tablets and put there, it gets healed, infection gets eradicated. And these sulfur drugs have a physical property. The physical property is it is not soluble in water. Since not soluble in water, pediatric preparation was not available. And one pharmaceutical company in USA found that it is possible to make a pediatric preparation by dissolving in another solvent which is similar to polyethylene glycol. And people are very happy, people have used this and see the result. More than 100 children died. More than 100 children died because of the kidney failure. Diethylene glycol, this solvent is polyethylene glycol. Realized. What is this realization? The drug alone is not important. The drug product is important. Drug alone is not important, but the drug product is important. And our pharmacovigilance includes the total product, not just the active ingredient. In spite of this, the American government could not even prosecute the company. The reason, there was no rule. Rule says the drug should be safe, but what about the product? Nobody talked about the product. And America finally prosecuted the company, not because of the safety issue, but because of the nomenclature, misleading nomenclature, because it was named as elixir. Elixir means it is a hydroalcoholic preparation, that's what we know from pharmaceuticals, but it did not have the alcohol. So because of this misnomer, company was prosecuted. Then coming to thalidomide, this is the disaster that everybody knows, and in pharmacology we teach. But many times what we don't teach is why this thalidomide was introduced and why it was an instant heat. Thalidomide was introduced as an alternative to barbiturate. barbiturate has a problem. What is the problem the barbiturate has? Safety margin is very low. One strip of barbiturate is enough to kill people. But thalidomide, even 100 tablets cannot kill a dog. See the high safety margin of thalidomide. The second advantage with barbiturate is, sorry, not with barbiturate, thalidomide. Barbiturate has a problem. If we take a phenobarbital tablet in the night, morning also will have the hangover syndrome. Thalidomide did not have. And that is the reason why it was an instant success. When it was instant success, it was used, and we see more than 10,000 children from 46 countries born with deformities and has the deformity. You know, a country, America. America did not have the effect of thalidomide digester. Whole Europe had, but America did not. The reason, you can see in the photo a lady standing in front of the American President Nixon. This American lady, an US FDA officer, medical officer, who was instrumental preventing the entry of thalidomide to America. So she prevented 2 years. How? You know, in America we have, if an application is submitted seeking approval, it should be disposed of within 6 months. If it is not disposed of within 6 months, it is automatically accepted as approved. This FDA medical officer, who was in charge of evaluating the dossier of thalidomide, in the last days of 6 months, returned back the application telling insufficient data. And that is how every 6 months he started doing, and she could prevent. And by the time this 2 years over, the information on phocomelia was out and people were afraid of thalidomide. This, sometimes people say, what we can do? You know, there was tremendous pressure in America to introduce, when whole world is using this medicine, why we will not use this medicine? And there was tremendous political and economic pressure on him, and she was firm on it. And America presented her the highest honor of America for saving the nation from thalidomide disaster. This is what happened in our country. 14 people died because of adulterated glycerin in the hospital, JJ Hospital of Mumbai. Court trial is contaminated with glibenclamide, killed many people. Diethylene glycol continued. Even sometimes back we had information that people died of diethylene glycol because of the ignorance. Some of you may not be knowing about this. I'll slightly narrate about the co-trimoxazole contamination. In Bangalore city, after the cataract operation, some people died. There was a lot of hue and cry in the press that all those who organize The ICAMs, blame goes to them. The doctors who operate it, blame goes to them. One of the young doctor said, if I do a mistake, if I do not properly take care of my patient will not die. My patient may lose the eyesight. Then only people's eyes are opened. They started investigating, and on investigation it was found that the medicine cotrimoxazole was given. For what? To prevent any infection. And this tablet contains glibenclamide. On investigation, they went to the factory, saw SOP and all, everything. And what they found is, while making the cotrimoxazole tablet, cotrimoxazole, a combination tablet, trimethoprim and sulfamethoxazole. While making this cotrimoxazole tablet, they used diluent also. And by mistake of putting starch in place of starch, that fellow has mixed the glibenclamide. And that is how the people who took this went hypoglycemia, and that led to coma and death of this. Look at the drug statistics. Of course, this is a 2005 publication which shows 130 products were withdrawn from the various market. 130 products were withdrawn from it. 1/3 is withdrawn within 2 years of launch. Very, very careful about the new medicine. And half is withdrawn within 5 years of life. New medicines always bring new surprises. You must be very, very careful while using the new medicines. And most of the time issue why they are withdrawn, either of cardiovascular complication, hematological or hepatic complications. The drugs are withdrawn. Next comes is how to teach. So there is a change in the teaching learning process throughout. It is no more a teacher centric. We all used to say he's a great teacher because he was a great speaker, and now the change, a great teacher who is a great motivator to the student, and the student learn themselves. That is the philosophy that we have at this moment. Not that I take a good class. How the students are learning. Yes, definitely is a satisfaction when I take a good class. People appreciate, but we are not stopping there. We are looking little beyond that. Yes, I have taken a good class. I have done my job satisfactorily. What about the students? Have they learned enough? And that is why, to make the learning experience more enjoyable, more effective, and more sustainable experience one get. And there are many student-centric methods are introduced throughout the globe. Experiential learning, integrated interdisciplinary learning, participatory learning, problem-solving methodology, self-directed, patient-centric, project-based. So some of the tools we should also use while teaching pharmacovigilance. Look at the regulatory aspects of pharmacovigilance. The Central Drug Standard Control Organization in our country, in association with Indian Pharmacopoeia Commission, is responsible for running this pharmacovigilance program. Schedule M of the drug and cosmetic rules requires serious adverse events to be reported to the licensing authority. All companies must have the pharmacovigilance system with an officer in charge, and the officer in charge qualified is specified either a medical officer or a pharmacist. Pharmacovigilance systems, when the master file, we have to submit to the Drug Control Authority, Licensing Authority. MA is Marketing Authorization Holder, means the company who received license is responsible for the pharmacovigilance program and periodically submit the data to the licensing authority. When we manufacture or import new drugs, this is a new rule, friends, new drugs and clinical trial rules in 2019. Those who want to import or manufacture should have a pharmacovigilance system. They have to submit 6 monthly periodic safety update reports, and there must have a mechanism for pharmacovigilance audit and inspection. Look at the pharmacovigilance system in this country. 2020 end figure. There are 311 centers, 37,731 reports it received, and quality of our report on an average or completeness of the score is 0.73 out of 1. It's a very, very high figure for a country like ours, when we have 57,731 report in 1 year. WHO Collaborating Center, IPC's Indian Pharmacopoeia Commission is recognized as a collaborating center for public health programs and regulatory services. What is the ultimate goal of pharmacovigilance, friend? To ensure rational and safe use of medical drugs, the assessment and communication of the risk and benefit, education and informing the patient so that the medicine can be appropriately used. As I said, friends, we have moved from drug safety to patient safety. Here are a few examples. Look at how the patient look at what is the patient expectation. Patient does not want needless death, so patient says, don't kill me. Patient says, do help me, don't hurt me. Don't make me feel helpless. Don't keep me waiting. Don't waste your resources as well as someone else's. So, the great medical philosopher. So harm comes, patient gets harmed from many sources, from many angles, and we are basically concerned medicine and medical products, how to make them safer for the users. Friends, initially the global fashion safety look at clean care is safer care, safe surgery is safe life, and in 2017 it has adopted medication without harm. Medication without harm, and we have most of the responsibility on preventing medication harm is on pharmacists. So those who want to take pharmacovigilance as career, they need to have competency, and what it says, Dr. Mary is not at this moment, is the director of Uppsala Center. She is the former director, who said the pharmacovigilance experts must have the observational skill of Sherlock Holmes, the great detective Sherlock Holmes. So that sorts of intelligentsia and observation skill is necessary. The then director, Dr. Mary said, when you have eliminated the impossible, whatever remains, however improbable, must be the truth. Friends, the biggest challenge for us is correlating the adverse effect with the drug, whether really the drug has caused these. So that is what she says. When you have eliminated the impossible, so it is a process of elimination, how this event has occurred and who is responsible for this event. When our people go for seeking a job to an organization, the interview board, owner of the company, director of the company, often ask, what can you do for us? Yes, you are all qualified, your CV shows you are highly qualified and you have achieved all the laurels, academic credentials. Tell me what you can do. Many times our students, prospective employees, they say, sir, whatever job is assigned to me, I can do. That means, we do not know what are the competency we have. We look at whatever job to do. Yes, whatever job the company assigns, we have to do. It is a need of the company once we become the employee of the company. But we must tell what are our expertise. So if it is a pharmacovigilance, some of this expertise, we must say, yes, I can identify the area. I can document it. I can analyze it, starting from the causality to the seriousness, serious adverse event, signal identification. I can do the data entry in the software. I can properly communicate with others on the adverse events. So here, friends, I just like to give you an example. The 2 children died because a wrong medicine was given when the children developed fever following vaccination. And usually fever is a common side effect of vaccination. So it is necessary to talk to the mother of the family or family members of the children, journalist, what action to be taken. You as a journalist, lot of questions people ask. And I will show you how 3 newspapers have reported differently. This was an incident of 8th March 2019. It happened actually 7th March 2019 and the newspapers of 8th March 2019. What newspaper headlines? 2 infants died, 22 taken ill after vaccination. Similar packing of tablets led to group of infants were given tramadol tablets instead of paracetamol. 2 babies died, 25 ill as health center gave wrong cure. Friends, when an ordinary citizen looks at these headlines, the ordinary citizen loses trust on vaccination. show trust on medicines, show communication about the pharmacovigilance or adverse drug. It should be a balanced way so that you tell the truth, at the same time don't lose the trust of the people on medicine. Friends, how communication can go wrong? Teacher asked this boy to write essay on dog, and he brought his dog to the teacher. So friends, coming to the artificial intelligence in pharmacovigilance, let me tell you, I am not a competent person to talk about the artificial intelligence. I am not trained on artificial intelligence. Yes, but I know about pharmacovigilance. So how we can use this artificial intelligence in pharmacovigilance? You may be hearing from other speakers, other learned persons who are very knowledgeable on artificial intelligence. But here, I just want to put you uh, a word of caution on artificial intelligence. Artificial intelligence, we are all experiencing artificial intelligence. The WhatsApp, when we start typing, some of the things automatically comes. This is artificial intelligence. When we are replying to a message in Google Mail, we write dear, immediately who has sent the mail, his or her name comes there. So this is all artificial intelligence. We all have been using it. So in the artificial intelligence, it has got two steps. One is, first, the machine, we are training them. We are giving a set of data from the past experience, and that is the learning phase for the machine. Then based on this learning phase, how the machine will react when we give them some work. So previously identify pattern that it has learned to indicate what something potentially helpful for us, and even it can also tell us whether an individual will or will not experience some adverse drug event. So this is one of very important area that artificial intelligence tells us. Artificial intelligence can also help us in assessing the case reports, large number of safety reports we have, and which are the serious adverse events. It helps us, and if we want to do, there could be error, there could be long time to do such analysis. So artificial intelligence makes the work easier for us. Friends, here is a recent happening that I would like to show here as a word of caution. Yes, we need to encourage our students to learn artificial intelligence so that their career prospects will improve. They will be in high demand because now artificial intelligence is everywhere. But there is a recent issue. The deepfakes are easy to create with artificial intelligence. Deepfakes are easy to create, and in order to prevent occurrence in our system, we need to invest heavily on technology If we do not invest, we will be susceptible to this new danger. United States oil companies paid huge amount to hackers to access their own data because the hackers have introduced a ransomware, and that prevented the oil companies from accessing the data in their own computer. It is your data, it is in your own computer, but you cannot have the access. So this is a word of caution while using the artificial intelligence. Friends, artificial intelligence cannot entirely eliminate errors. So let us implement this to enhance, but not to replace the current work process. Thank you very much for all patient hearing, and please feel free if you have some concern, some questions. I shall be very happy to reply. Before I close, yes, we have all this time we have talked about the ill effects of medicine. We have talked about the problem associated with medicines, but medicines do wonder to us. So here is a small joke. Enjoy it. Don't take the other way. And pardon me if it hurts someone sentiments. I have training on gender bias. I have training on gender sensitivity, and there is nothing with any motive. It is just to enjoy. Friends, girls, girls, girls. If you appreciate, they say you are lying. If you speak, they say you are not listening. Hello? If you speak, they say you are not listening. If you speak, you are not caring. If you kiss, you are not polite. If you do not kiss, you are fit for nothing. This is what the wonders we listen. Girls, girls, girls. It is difficult to live with them. It is difficult to live with them. Friends, but more difficult to live without them. So is our medicine. Yes, medicine have all these issues, and that is why we have pharmacovigilance as a new discipline. But medicine has all these issues, but we cannot live without medicines. Take home message: no medicine is 100% safe. It is understandable that the patients die of lack of access to medicines, but it is not acceptable the patients die of medicines. This is a noble and satisfying area to work, pharmacovigilance. Let's ensure the medicines available for us as well as for our future generation safe and effective. Friends, this is the reference. Most of the things that I discussed today is from my book, Textbook of Pharmacovigilance, Concept and Practice. Thank you very much. Any question, please?
Well, participants, if any questions are there to be discussed with Professor Mohanta sir, Kindly you can unmute yourself and you can ask your question. Sir is there to answer you.
Friends, you are all teacher. You know best what to teach and what not to teach. This is just sensitizing most of the young teachers.
Yes, sir. Okay. Sayate?
You
Yes,
can
sir.
even post, please. You can even
Yes.
post the questions. Our Satili has already put in the chat box.
Yes, sir. Yes, sir. So, participants, if you do have any questions to be discussed or answered, kindly share it in the chat box, please.
Great, we have 1 century mark, 100 participants.
Yes, sir. Yes, sir. Okay, sir. Sayate, you can propose a word of thanks, ma.
Yes, sir. So, thank you very much, sir, for being with us and for sharing the knowledge and how the pharmacovigilance and how the pharmacists are involved in that and how the AI can be helpful for us and what are the harms the medicines can do and how we can helpful with that. And we are very honored to be listen to your session, sir. Thank you so much for being with us.
Thank you, Mohanta, sir. I'll get you back, sir.
Sure, sure.
Thank you. See you,
Most
sir.
welcome.
Yeah. Participant, you can join for the very next section by 11 o'clock. The link is already shared in the WhatsApp group, and also it been shared in your email, registered. But, sir, there is a question, sir.
Yes, yes. As
Yes.
pharmacovigilance is taken vital role in drug safety monitoring, but how it is applicable in drug repurposing?
Yes, sir. Drug repurpose.
Repurposing. Friends, very interesting question you have asked. The repurposing, when we are using for some other purpose other than it is indicated, and this mostly we see here during this COVID-19, because when we talk about the COVID-19, people say the doctor do not know, the scientists do not know. Yes, how people will know? The virus itself is new. This virus did not exist prior to this COVID-19 period. So naturally, we do not know. It is like little bit of whatever idea we have. From that, we think, okay, this drug may be useful. Like that, that is why people have been using. And when we are using the drugs, repurposing its drug is definitely we have to see, because the drug is indicated for something else, but now we are trying testing it. Repurposing means testing it in another disease, another condition, and the drug safety monitoring is very, very important, and it must be a part of it. Whenever these see these repurposed molecules and all, they give emergency authorization. It is not the full authorization, full marketing authorization, but emergency authorization. And emergency authorization is always as a component of drug safety monitoring.
Participants, if you do have any other questions to be discussed, kindly post your questions in the chat box. Sir is there with us to share his expert and knowledge. Please, if any questions are there, kindly post in the chat box, please. Okay, sir. I
Okay,
think
thank you.
there is no questions are
Uh,
there. It was very
Thank you very
clear.
much.
Thank you, sir.
Uh,
Thanks a lot, sir, for joining
Thank you
us.
very much. Thank
Thank
you.
you, sir.
Thank you, sir. Thank you so much for joining us. Is respected participants, the next section will start by 11 o'clock. So please be join at that time. And a small reminder again, while filling the feedback form, please mention your participant ID very clearly. There are few mistakes. If there are any mistakes, it will not be considered as attendance. So please check your participant ID. It has already been shared to you. So please fill the participant ID correctly while filling the feedback.
Ma'am, excuse
Please
me.
join by 11 o'clock
Yes.
again.
Yeah, participant ID was in that PDF format, no, ma'am, which you have
Yes,
shared.
yes, sir. Yes, sir. Yes.
Okay, thank you, sir.
So it is there like your registration ID, your name is there. So what whenever you are filling the form will be asking you your registration ID, and you have to give your ID. Like the ID will be like 1904 AI 0010 or 11. It will be like this. All right. Then only the system will be accepting your responses. Okay.
Okay, sir.
Thank you. Thank you. We'll meet for the next section by 11 o'clock by Dr. G. Narayan, sir, on evidence-based medicine and artificial intelligence. Thank you all for joining us.
Thank you, sir. And the
Okay.
link has been shared in the WhatsApp group. So please join to that with the particular link, respective link. Thank you, everyone.
Thank you. Dear participants, kindly you wait for 2 more minutes. Uh, the participants are joining. All right. So bear with us for 2 more minutes. We'll be starting. Savitri,
Yes, sir.
I think we can start.
Yes, sir. Good morning, participants. Welcome back to our session 2 on day 2. So, for this session, we are having our speaker, Dr. Goranta Narayan, sir. I'm very pleased to invite our speaker, Dr. Goranta Narayan, sir. Sir has received his Doctor of Philosophy from Jawaharlal Nehru Technological University, Anantapur, and received his degree in Bachelor of Pharmacy and Master of Pharmacy from Annamala University, Tamil Nadu. So, after completing his graduation, sir has joined Raghavendra Institute of Pharmaceutical Education and Research as assistant professor, and currently sir is head Department of Pharmacy Practice, Raipur Autonomous, and sir is a hospital in charge since 2009, and sir is scientific coordinator of ISPAR, International Society for Pharmacoeconomics and Outcome Research, Andhra Pradesh, AP Regional Chapter in February 2009, and director of ISPAR AP Chapter since 2015. And sir is having various expertise in the areas of pharmacoepidemiology, economics, clinical epidemiology, community medicine, and interested in pharmacovigilance, therapeutic drug monitoring, medication therapy management services, patient counseling, and community educational programs. And sir has organized many conferences, symposium in RDT Hospital, Batalpalli, and in Raipur. Sir has recognized as supervisor, guide for undergraduate and postgraduate students, and has published more than 50 papers in national and international journals. And Sir is having a vast interest in pharmacopepidemiology research activities. I hope we can learn a lot from our next speaker. With this brief introduction, I'd like to invite Dr. G. Narayan, sir, to start the session.
Okay, thank you very much, Sahiti. I think my screen is visible to all.
Yes, sir.
Thank you very much. Firstly, I would like to thank All India Council for Technical Education and Raghavendra Institute of Pharmaceutical Education Research for giving this wonderful opportunity to share my knowledge to the faculty community. Today, I am going to discuss about fundamentals of evidence-based medicine and application of artificial intelligence in clinical decision making. If we are going to look overview of the EBM and artificial intelligence, the primary objective is one, to make a right decision to improve the patient outcome. For understanding purpose, I had made this session into 3 parts. The first part, we are going to discuss about what is meant by evidence-based medicine and what is the necessity for the EBM in the current clinical practice. In the second part, we are going to discuss about what are the different steps involved in the evidence-based medicine. And in the third part, we are going to look upon how artificial intelligence is going to be applied in clinical decision-making process. Especially, EBM will be having certain gaps, how the artificial intelligence is going to fill that gap, that we are going to discuss in the third part by using a evidence-based examples. Okay, right? These are the contents which we are going to discuss today. Right? In routine clinical practice, majority of healthcare workers or healthcare professionals, they will be having so many questions about the management of the patient. Like, shall I order this test to my patient or not? Or what type of drug I need to select for my patient? Or if I am going to give this drug, how much magnitude of the effect is going to be expected from the patient? These are the routine questions which are going to be arranged in the practice. The answer for this question is going to depend upon two factors. One is physician perspective and another one is patient perspective. If we are going to see on the side of physician perspective, the factors like knowledge and their skills, expertise, and even what are the resources available for the physician, it is going to decide what type of decision they are going to take. Equally, not only physician perspective, parallelly, even patient expectations or patient perceptions are also very important and crucial to make the decision to recommend that intervention or to stop that intervention. EBM is a tool which is going to consider both the physician factors and patient factors to make a right decision. EBM is going to provide a strong research evidence which is going to implement into the clinical practice. We will see that what's the process will go on in the EBM. Okay, right. Coming to the definition of the evidence-based medicine, I hope everybody is familiar for this definition. The definition was coined by David Sackett, who is the British epidemiologist. He is the first person who made the clear definition for the evidence-based medicine. On the screen, you can observe three definitions. I personally recommend the third definition. If you are going to observe the first one, it is going to indicate that explicit and judicious use of the best research evidence in decision-making process. Whereas if you are going to observe the second definition, integration of the best research evidence and clinical expertise in the decision-making process. In both these two definitions, we didn't consider either patient values or clinical expertise. So that the third definition, I am personally going to recommend that, which was coined by the David Sackett, integration of the best research evidence with clinical expertise and patient values. Okay, where we are going to consider all the three domains, not only best research evidence, even equally the clinician need to incorporate his skills to take the decision, and even he should consider the patient perception and values also. It is also very, very important. Let us see how the evidence-based medicine relates with the other branches. You can observe on the screen, there are so many other branches are there, like clinical pharmacology, evidence-based practice, molecular biology, clinical chemistry. The primary core value for these branches is the outcome of the patient is going to depend upon the mechanism of the drug. This is the primary core value for these branches. Okay, like if we are going to see the evidence-based medicine, it is apart from all these branches. Even these branches are going to criticize the EBM, like EBM is empirical. In real scenario, it may work or may not work, and even they are going to criticize that EBM does not have any strong scientific background to promote into the patient outcome. Okay, these are the critics from the other branches. If we are going to see the on the side of the EBM, as we well know about that, the outcome of the patient is not only depends upon the mechanism of action of the drug. There are various factors are going to influence the outcome of the patient, like it may be age or genetic structure, gender, and comorbid conditions, and what are all the concomitant medications taken by the patient, and severity of the illness. These are all the various other factors which are going to influence the patient outcome. Here, the EBM is going to provide a key by considering all the factors. It is going to judge whether the patient is going to get a better outcome or not. Okay, that is the highlight of the evidence-based medicine. Right now, let us move how the EBM is really practical. It is very important. The three alliance like research evidence, clinical expertise, and patient preferences. These three alliance is very important to make a right decision to get a better outcome. If we are going to see on the research side, whatever the research that may be related to the prognosis, or that may be related to the diagnostic, or it may be related to the interventional study, the research it should be a clinically relevant research, and it should be patient centric. Then only we can able to consider that it is going to fit for our decision making process. Coming to the clinical expertise, it is very important. The clinician need to use his expertise or skills to assess whether that evidence is really suitable for their patients or not. It is very important. He must in the competency, like he can able to measure what would be the benefits for the patient or what would be the risks for the patient if he is going to recommend that intervention. That expertise is very important, not just taking the evidence and implement into the practice. It does not going to work out. They need to use their expertise in making a right decision. And it is also very important that we need to consider what are all the expectations of the patient. What are all the values, what are the perceptions of the patient. It is very important. If these three are going to having a strong alliance between these three, we can definitely achieve the right decision. We are going to achieve the right outcome. Now let us move to why evidence-based medicine is required in the current clinical practice scenario. If we are going to observe the research and practice, there is a wide information mind gap is there between the research and practice. If we are going to see on the side of the research, majority of the researchers, they are unaware about what actually the practitioners require. Okay, and one more thing is, even sometimes the researchers well known about what the practitioner required, but they are going to publish their work which is not accessible for the practitioners. Coming to the practitioner side or practice side, a huge amount of the research is publishing on day-to-day basis. It is very, very difficult to update the research on day-to-day basis. Okay, and even majority of the practitioners, they are unaware about what to update, what not to update. And even if they are having interest also in so many conditions, they don't have right resources or tools to access that right evidence into the practice. Okay, so that there is a wide information gap is existing between the research and practice. EBM is going to fill this gap and provide right evidence to implement into the practice. That's why we need EBM, right? As I said that there is so much of evidence, but we are having less time. Just you can observe how much of literature is publishing on day-to-day basis. Okay, these are the articles which are PubMed indexed journals, and that too Medline indexed journals. For a practitioner, if he want to update the evidences on day-to-day basis, you can observe that only randomized control trial around 50 per day are going to be published. If any practitioner want to update each and every article, he need to read each article in half an hour. That is going to indicate that it is very impossible. It is very impossible. It is not possible. So that EBM is required to solve this problem. Right. If we are going to observe the research which was conducting in global level, we can divide that research into 4 quadrants based upon the applicability and based upon the advancement of the knowledge. Like 4 quadrants, we can divide that research into 4 quadrants. One is Curie quadrant, Pasteur quadrant, Dahl quadrant, waste quadrant. Coming to the Curie quadrant, the research is purely basic research. It does not have any practical applicability. That's what we are going to call as a Curie quadrant. And coming to the Dahl quadrant, it is purely applicative research. Means like some certain problem aroused in the practice, we are going to conduct a small research to get a solution for that problem. And we are going to solve the problem. Again, one more problem will come. Depending upon the problems, what we are going to face in the clinical practice, we are going to conduct the research. It does not have any benefit to advancement of the knowledge. This is what we are going to call as a Dahl quadrant. And the main quadrant is Pasteur quadrant, the research which is going to advance the knowledge, and as well as it will be having high practical applicability. The one quadrant is what we are going to call as a waste quadrant, which does not have any practical applicability and it don't have any advancement of the knowledge. These are the 4 quadrants. This is going to indicate that what type of research we are conducting. Okay, we need a Pasteur quadrant type of research which will be having high applicability and which is help to advance our knowledge to face the future problem. It is very important. Right? Now, I want to discuss a real historical examples which will show the information gap between research and practice. Hope everybody knows that usage of the corticosteroid therapy in preterm birth to prevent preterm birth complications. I hope that everybody knows that, like now even in WHO guidelines also it is mandated that if any pregnant woman expected to deliver in preterm birth, okay, it is mandated to give dexamethasone or betamethasone 4 doses by a 24-hour interval. It is a guideline which is available. Okay, the corticosteroid therapy is recommended. Why? Because preterm birth is associated with more birth complications like birth asphyxia, lung immaturity, neonatal sepsis, neonatal jaundice. These are the major complications. Low birth weight. These are the complications which are associated with the preterm birth. If we are going to see the evidence, in 1972, the first randomized controlled trial was reported on usage of corticosteroid therapy can prevent the preterm birth complications. Similarly, in 1972 to 89, 6 more randomized controlled trials are published supporting the first evidence. Even in 1989, the first systematic review was published on usage of the corticosteroid therapy in preterm birth to prevent the complications. I hope that this is the first systematic review. If you are going to observe the Cochrane logo, the forest plot is going to indicate the corticosteroid therapy study review, systematic review. Okay, this is the high level of the evidence which is helpful for the clinical practice. From 1989 to 91, 7 more studies are reported that corticosteroid therapy is going to be having beneficial effects if we are going to use it in the preterm birth. The evidence is very clear that if we are going to use the corticosteroid therapy, it is going to reduce the deaths associated with the preterm birth. Okay, it is indicated that corticosteroid therapy will reduce the odds of dying of children due to preterm birth to around 30 to 50%. The evidence is very clear, but unfortunately, majority of the practitioners are unaware about what's the evidence is existing, so that the disaster may happen. Like they are not using the steroids, and so many neonatal deaths we faced. This is a wonderful example which indicates that a gap is existing between the research and practice. One more historical example which indicates a clear gap between the research and practice. I hope that everybody know Bernard Lawn. He is the cardiologist, famous cardiologist, and also he is the person who invented defibrillator, got a Nobel Prize for the invention of the defibrillator. He addressed in one conference which was organized by the American College of Cardiology. He made a statement that the most common cause of deaths in middle-aged people is heart attack. Before heart attack, the person is going to experience the supraventricular tachycardia. If any scientist, if any researcher is going to invent the drug, which is a long-acting antiarrhythmic drug, it can probably reduce the deaths associated with the heart attack. Okay. After this address, so many researchers and scientists went for the research, and finally, one publication we got about usage of the oral flecainide acetate for the treatment of ventricular arrhythmia, which was published in the New England Journal of Medicine, which is a topper, high-impact journal in the medical field. I think around 45 impact factor is there for this journal. Okay, the study indicates that these are the results which got from that study. It is very clear that it is a crossover trial. On first assignments, the placebo group shown more number of preventricular contractions, whereas in flecainide group, the patient showed a suppression in the preventricular contraction. After crossover, the same thing was repeated. In the flecainide group, less preventricular contraction. In placebo group, there are more preventricular contraction. It is very clear that the flecainide group, flecaine's drug is going to reduce the preventricular contraction and suppress the arrhythmia. Thereby, we can prevent the heart attack, or we can prevent the future occurrence of the heart attack. This is very clear. The statement is very clear. It was got wide publicity. Majority of the cardiologists or general medicine practitioners, they trusted the resource. Why? Because NEJM means it is going to be having more trustworthy information. It got more popularity, and they started the usage of the flecainide in the treatment of the heart attack. After a few years, one more publication released on flecainide. This was published in the Circulation. It is also high impact journal. The trial is CAST arrhythmia suppression trial. The thing is, in this study, they observed for a long period of time, and to the conclusion that in the flecainide group, Unfortunately, these findings popularized. Practitioners or the cardiologists are evidence. They are giving the flecainide drug to prevent the, and they are systematically. This is the gap is there between the research and practice. That's why to select a right evidence and to implement that evidence into the practice. Evidence-based medicine. What are the different? There are 5 steps are there. One is formulate an questions, but we can't type complete format or Cochrane or Embase. We can't type the complete query. We need to we can get a right evidence. Means during our search, we should not miss any evidence which is relevant for our patient. And at the same time, we should not get searching. That is the primary motto. Complete important. And the second one is a lot of evidence will be there. We need to systematically track the best by using the search engine. Okay, we are going to get the evidence, whether directly we are going to implement that evidence into. We need to validate that guidelines are there depending upon type of design. If it is observational study, depending upon the study design, we are having checklist to evidence and whether it is applicable for our patient or not. We are going to check it. Everything, we are going to apply that evidence into the practice. After application of the evidence into the practice, our job is not going to be completed. The outcome was improved compared to the previous practice. Continuous manner. These are the 5 very, very important steps. It is a heart for the one by one, right? The first one is formulate an answerable question. Whatever the query you are having, firstly, question and reconstruct that question into the appropriate. We are going to use is we need to divide that query into PICO format. P means population, I means intervention or diagnostic test, whatever the intervention it may be. Okay, we need to clearly mention that intervention. And C means comparator. It may be either placebo or it may be a standard drug. And O means what are all the outcomes. Most of the time, if we want to get evidence, means in majority of the cases, we are going to use the population and comparator to get a complete relevant research. If you are more specific about the outcome, we need to mention outcome also. Okay, I hope that you understand the PICO format. I will give one example. Later, I will check how much you understand from that example. This is the query. Jeff, a smoker of more than 30 years, he tells you he has tried to quit smoking, but he unsuccessful in the past. Whatever the techniques he tried, he is unsuccessful. A friend of his, however, successful, quit with acupuncture. Okay, by using acupuncture, we got a good outcome. Should he try it? Okay, that's the query. Other interventions which are available in the market are either going for nicotine replacement therapy to overcome the withdrawal effects, and antidepressants are available to cope the stress after quitting the smoke. These are the other interventions which are available in the market. Now we will see how to frame this complete query into answerable format. This is the intervention question. Like this. The population is smokers, and what intervention we need, what the evidence we need means how the acupuncture is going to help in quitting the smoking. And the comparator is antidepressants or nicotine replacement therapy. We can keep the comparator. If you want a comparative study with the existing strategies means we can keep comparator. And the outcome is quit smoking. Okay, right now I will give one query for you. You need to answer this query. Who is the population? What is the intervention? What is the comparator? And what is the outcome? Okay, I hope that you are able to see my screen. I am unable to see you. Okay, right. This is the intervention question. In browsing one of the medical weeklies, you come across that imiquimod cream for treatment of basal cell carcinoma. Okay, it is the idea of a cream for the basal cell carcinoma is surprising for you. So you wondered about that, whether it is work out in the basal cell carcinoma or not. That is the query running in your mind. Okay, now you want to go get an evidence about imiquimod cream, how much it is effective in long-term cure rate in the basal cell carcinoma. Okay, I hope that you got the query. Just I want to see your answers. That's why I am opening the chat box. Okay, you can type what is the population, what is the intervention, what is the comparator, and what is the outcome. Please, participants, you can type in a short answer. No need of a big answers. You can try in a short format. Anyone? Okay. Without running our time, like anyone, you can please, if you got understanding about how to convert our query into the answerable format, you can type what is P, what is intervention, what is comparator, and what is outcome. Okay, or otherwise, any person can unmute your microphone and you can express your answer if you are having time consuming that typing the answer means. Okay, good, no problem. Okay, I will run the presentation, right? Okay, this is the question, right? If you want to what's the answerable format for this query means, right? The population is patients who are suffering with basal cell carcinoma, and intervention means imiquimod cream, and the comparator is what are the conventional methods which are available for treatment of the basal cell carcinoma, and the outcome is long-term cure rate. This is the way how we need to structure our question based upon our query. And hello, I think some disturbance is. Participants can mute your microphone. Right. Right. One more query. George has come into our surgery to discuss the possibility of getting a vasectomy. He says he has heard something about vasectomy causing an increase in testicular cancer later in life. You know that the risk is very low, but you want to give a precise answer for him. Okay, you want to search that if the person will undergo vasectomy, what would be the likely risk to get a testicular cancer? Okay, this is somewhat related to the risk factor question or etiology question. We can frame that vasectomy means population is adult males and intervention is vasectomy and the comparator is no vasectomy and the final outcome is the risk of getting testicular cancer. Okay. And one more question. Susan is expecting her first baby in 2 months. She has been reading about the potential benefits and harms of giving newborn babies vitamin K injection. She is alarmed by reports that vitamin K injection in newborn babies may cause childhood leukemia. She asks you if this is true, what is the risk for her baby? Okay. And the answer is, how to frame this means the population is newborn and intervention is vitamin K injection and the comparator is in the babies where we are not going to give any vitamin K injection and outcome is leukemia. This is the way how we are going to frame our answerable question. And this is the last query. Julie is pregnant for the second time. She had her first baby when she was 33 years and had amniocentesis to find out if the baby had Down syndrome. Okay. The test was negative, but it was not a good experience as she did not get the result until she was 18 weeks pregnant. You see that one of the diagnostic tests to find out Down syndrome during the pregnancy is amniocentesis. Unfortunately, the result came 18 weeks after pregnancy. Okay, if she want to make any decision about continuing the pregnancy or in certain cases, these results are not going to satisfy. After 18 weeks means it will be again complicated. Okay, now she is 35 years, 1 month pregnant, and asks if she can have a test that would give earlier result comparing to the amniocentesis. She want to take the decision like before itself. The local hospital offers serum biochemistry plus ultrasound screening as a first trimester test for the Down syndrome. Now you want to know whether this intervention like serum biochemistry plus ultrasound screening is going to give accurate diagnosis of the Down syndrome compared to the amniocentesis. This is the query actually. How to frame this question means pregnant women, intervention is serum biochemistry plus ultrasound screening, comparator is conventional amniocentesis, and the outcome is accurate diagnosis of Down syndrome. Okay, this is the way how we are going to divide whatever the query we are having in our mind. We are going to segregate that into an answerable format by using the PICO. Okay, even if you will go through Cochrane library also, they are going to give these options P, I, C, O. What is your P? What is your I? What is your C? And what is your O? Okay, we should know that is a basic to start the EBM process. Right? We completed the first step, and now we want the evidence. Okay. Firstly, we need to know what type of question is there, like whether we want to know about risk factor, or we want to know about burden of the problem, or we want to know about treatment efficacy, we need to very clear about our question. We should not just directly jump into the searching process. We need to clear about that. Depending upon our question, the type of study design which we need to take, it is going to be changed. I will give a wonderful example. Like recently, everyone is in pandemic situation, COVID-19. I will take that example only, because everyone is familiar for that example, right? Just go back to 1 year past, like in 2019 December. Consider that you are a general practitioner working, and in one new case you got that you never heard about that case. Like the symptoms are very different. Like they are telling loss of smell, loss of taste, and continuous cough, fever. Okay, this is somewhat a new type of symptoms the person is getting. Okay, now what type of design we need to select? Okay, it is nothing but you are observing all those findings in the single person. Okay, in such a case, that design will be a case report. Okay, then the same findings of the patients you are experiencing in your clinic. Okay, you are getting a data from a 10 or 5 patients who are having the same symptoms. Okay, then that condition we are going to call as a case series. When compared to the case report, case series is higher level of the evidence. Similarly, like you know about what are the findings, what are the symptoms, certain population are getting those symptoms. Okay, it is very clear. Now you want to identify what is the problem, why these people are getting such type of symptoms. You want to know what is the risk factor, what is the cause, what is the etiology, whether the person is getting through water contamination or through air or through any other mode, contaminated food. Why these people are getting such type of symptom? You want to know what is the risk factor for that condition. Okay, then you are going to take a study design called either case control study or cohort study. Okay, and you want to know what is the burden of the problem in your residing area, or it may be in the district level or state level. How much burden is there? How much prevalence is there? Okay, in such a case, you need to go for the cross-sectional study. Okay, and now you are having you literature that remdesivir or certain drugs are showing a promising results in the previous pandemic conditions like Ebola or H1N1 virus. Okay, now you want to test that whether they are going to work out in the COVID-19. Okay, now what type of research you are going to do? Means what type of design it should be? Means randomized control trial. Like that, depending upon our question, the type of evidence what we are going to take, it is going to be changed. Okay, if we are going to see the hierarchical levels of the evidences, the topmost evidence is 1 minute. Wait a minute. Okay, the topmost evidence is systematic review or meta-analysis. Okay, the lower level of evidence is case report or case series or expert opinion. Okay, this is the levels of evidences: case report, case series, cross-sectional, cohort, case control, RCT, and in RCT again open labels, single blind, double blind, triple blind, and then combination of all the RCTs, which is going to give the topmost evidence, what we are going to call as a systematic review or meta-analysis. So finally, from this systematic review, we are going to develop our guidelines. Why? Because if any organization or hospital authority or policy making persons, if they want to frame the guidelines, means going through each and every RCT is very difficult task. So that they will take a systematic review, and they are going to incorporate that evidence into the clinical guidelines. This is the way how the evidence will flow. Okay. Now let us see how to retrieve the best research evidence. Okay. Like everyone, I think you are more familiar about Boolean words when we are going to use search either in PubMed or Cochrane. Basically, we are going to use three Boolean words. One is or and not. So these are the three most commonly used Boolean words. And by using these Boolean words, we are going to type our PICO whatever the question you are having in the search engine, then you are going to retrieve the evidence. Okay, right. Depending upon the type of question, where you need to search. Okay? If your question is related to the intervention, okay, don't go directly to the PubMed. First, you search in the Cochrane library. In Cochrane library, if you are going to find any Cochrane systematic review, like it is a Cochrane collaboration is a non-profit organization which is going to synthesize the evidences in the form of systematic review or meta-analysis by taking the available evidences in the global level, and they are going to give the systematic review which are going to be helpful for the practice. Okay, so that it is better to go firstly if you are going through the Cochrane library, you are going to find your best evidence. Okay, if you are going to find any Cochrane systematic review, you can stop your search and go for the critical appraisal. If you didn't find any systematic review, then you can go for the DARE database. DARE means database of abstracts of review of effectiveness. It is a DARE database. In DARE database, if you are going to find any systematic review, you can stop there and go for the appraisal. If you are unable to find systematic review, means you can go for another option will be there. This is what we are going to call as a CRCT, Central Register for Control Trial. In this database, we can find randomized control trials. If you are going to get randomized control trial, you can stop your search and go for the appraisal. If you didn't find any randomized control trial, then you can shift your search to the PubMed. In the PubMed, the same process will go on, and if you will get a right evidence which is relevant for your patient, you can stop your search and go for the appraisal. Okay, this is the systematic way how to search and get a right evidence. Like as I said that we need to use Boolean operators during our search. Like for example, if you will see, if you want the evidence related to the colorectal neoplasm and related to the screening, and you want to add mortality, means you are going to use AND as a Boolean operator. Okay, means if you are going to use mortality, capital AND, screen, capital AND, and colorectal neoplasm, then you are going to find the research which is going to having all these three terms. All these three terms. Okay, and if you are going to use Boolean operator like capital OR, mortality, capital OR, colorectal neoplasm, you are going to find the articles which may contain mortality or colorectal neoplasm. Okay, not means it is going to exclude the term from the search. Okay, like that, these are the Boolean operators which we are going to use in our search strategy to get a right evidence. Okay. The next step is appraisal of the evidence. It is very, very important step and crucial step in the EBM process. Everything is going to lie in this area only. Once if you are going to appraise that evidence, you will go and discuss with the policymakers or the administrative staff about that intervention, what are the strengths are there, what are the weaknesses are there about that intervention, and what is the cost, everything. All these discussions will go on, and finally you are going to take a decision that to implement that evidence into the practice. Okay, for understanding purpose, I am not going to touch all types of study design. Okay, I am going to discuss about the randomized control trial, how to appraise the randomized control trial before implementation into the practice. Okay, it will be validated by using two modes. One is internal validity and another one is external validity. Internal validity is going to talk about methodological issues, whatever existing in your research that we can find in the internal validity. Whereas in external validity, it is going to talk about how much the research is relevant to your patients, whether the outcome which you got in that research, the same we can expect from your patient. All those things we are going to talk about in the external validity. Okay. In internal validity, majorly we are going to find what are the chances for the risk of bias in the study. We are going to look upon over the methods and what is the possibility of entry of the bias in that study we are going to analyze. Okay, risk of bias is nothing but situations where the result of the study does not accurately represent the truth. Okay, that we are going to check in the internal validity. Okay, right. Let us see what are the sources of bias in the randomized control trial and what type of parameters we need to check in the article. Whenever you are going to appraise the evidence or appraise your article, whatever the collected article, what are the areas we need to check and what parameters we need to consider in the bias assessment. There are 5 areas where the bias can introduce into the study. One is bias arising from the randomization process, what we are going to call as a selection bias. And the second area is bias due to deviations from the intended interventions, means the interventions whatever we assign for the patients, if they are not going to be similar, it is going to introduce the bias. That bias we are going to call as a performance bias. And the third area is bias due to missing outcome data. It is very important. Like sometime you are going to calculate the sample size by using predefined parameters, and you are going to run your trial. After completion of the trial, there will be certain withdrawals will be there. Certain data is going to be missing from the patient. Okay. In such a case, even the bias is going to be arise due to missing of outcome data. This bias, what we are going to call as a attrition bias. And the fourth area is measurement of the outcome. How we are going to measure the outcome. Whoever the person we allocated to measure the outcome, if they made any partiality in the measuring the outcome, it is also going to introduce the bias. That bias we are going to call as a measurement bias. And fifth area where the bias is going to be possible means during reporting of the results. Okay, you completed your study and then you want to publish your study in a well indexed and high impact journal. You will go for the publication. During publication, you will make selective reporting to that journal. That is also going to introduce bias. That what we are going to call as a reporting bias. Let us see one by one. What are the parameters we need to consider in each area where there will be a chance of the bias? Okay, right? In case of selection bias, what we need to observe in our evidence means Was the method of randomization is truly random or not? We need to check that. And the second one is, was allocation after randomization, we are going to allocate drug to the test group and standard to the control group. That allocation is concealed or not, we need to check that. And the third parameter is where groups are comparable at starting point of your study. It is also very important. At starting, the baseline characteristics of the test group and the control group, they are matching or not. We need to know. If they are not equal, means the outcome what we are going to expect, that is not going to be due to the drug, and even the confounders are also going to involve in the alteration of the outcome. So that these are the three parameters which we need to consider to reduce the selection bias. Okay. Right, these are the modes how to reduce the selection bias. When you will go for the randomization, it is suggested that to go for either random number table or play cards or coin, and the best mode of randomization is computer generated random number. It will give high probability in getting the random numbers. Okay. Like we can't guess in case of computer inserted randomization process. Okay. And even concealment is very important. Okay. Most of the time we are going to allocate the drug in envelopes. Okay. There will be chance for breakdowning of that envelope, like whether it is test group or control group. Then it is better to go for the computer allocation concealment. Okay. And the next one is performance bias. Okay, what we need to observe in performance bias means where patients and practitioners providing care, they really blinded or not. Okay, like single blind, double blind, triple blind. It is very important. If we are going to blind, they will treat both the test and control in similar pattern, and the outcome we can expect from only drug, not due to the person involved in the test or study. Okay, and the next type of bias is measurement bias. Which parameters we need to consider means we need to check that whether that outcome, whatever they framed, it is relevant for the patients or not. Okay, sometimes the study may have primary outcome, secondary outcome. There will be so many outcomes, but when coming to the result, they are going to discuss only few results, partial results, but they are going to left other things. Okay, but we need to clearly observe what outcome they framed, whether it is achieved or not, okay, whether it is relevant to the patients or not. That we need to check. Okay, and it is also very important that whatever the test they had used to assess that outcome, whether it is valid or reliable or not. Sometimes the outcome maybe can be assessed by using some formats of questionnaire, by interview based. Okay, it's very important. Okay, whether that outcome, what the mode of assessment, whether it is relevant for that patient or for that condition or disease, we need to check that. And more important point is sample size calculation. Okay, the sample number, it has a sufficient power to assess the difference between the group, that we need to check. That is very, very important. Okay, if it is a less sample size and it is showing that there is a significant difference, there is no significant difference, means we can't trust that result. We can't expect that it is going to be happen. Okay, so that it is very important whether that sample is powered to detect the outcome or not. Firstly, we need to check that. Okay, these are the things which we need to consider in case of measurement bias. And the next one is attrition bias. As I said that initial sample will be some number. After running of the study, there will be loss to follow up. There will be so many things will be there. So that we need to check that whether the attrition bias is there or not. If there will be any attrition bias, whether they conducted intention to treat analysis or not. Like for example, there are 50 numbers at the starting stage of the study, 25 in test group and 25 in control group. Okay, after completion of your study, you lost 10 patients in the test group and 5 patients in the control group. These losses, whatever the loss is there, even after loss also, the power is maintained or not. That is very important. Okay, that also we need to check. And generally, this is the rule what we are going to use for loss to follow up. If there is 5% loss, then we can consider that as a little bias. 5 to 20% of the initial sample size, then it is going to be called as a small bias. More than 20%, definitely we need to consider that as a serious threat to the validity of the result. Okay, it is also very, very important. And the last one is reporting bias, very important. At starting point of the study, the researcher is going to plan so many outcomes for his study. Okay, but at the end of the study, when he will go for the publication, he will publish the work what he got as significant results. He will left the non-significant results. Okay, this may also going to introduce the bias that what we are going to call as a reporting bias. As a practitioner, we need to check that whether they completely reported in the publication or they gone for any selective reporting. How we can track this thing means any study, if they are going to conduct, they must register their trial. Okay, for example, if you will take in our country, Clinical Trial Registry of India, CTRI, and if we are going for the global level, clinicaltrial.gov. Okay, there are registry is very important before start of the trial. Okay, even if you are going to take systematic review also, who are planning for systematic review, they need to register their protocol in Prospero. Okay, there are certain databases are there. We need to register that. Okay, like what's the necessity for the registration means we are conducting the study and it is transparent to the all the scientific community and public. Okay, like as a practitioner, if you are having any doubt about the reporting bias, we can track the protocol and we can see that what are all the outcomes he selected at the initial stage of the study, and at the reporting stage what he mentioned. By comparing this, we can get a clear idea about whether there is a reporting bias is there or not. Okay, this is the way how we will do the internal validity to address the methodological issues which are going to present in the study. And the second one is external validity. The thing is, whatever the findings we are observing in the evidence, whether those findings we can expect from our patients or not, that we are going to address in the external validity. Okay, for a simple understanding, I am giving the same steroid usage in the preterm birth. This is Liggins study. By observing the 2x2 table, we can make certain conclusions. Okay, like in corticosteroid group, 36 neonatal deaths are there among 532 births, whereas in non-corticosteroid group, among 538 neonats, 60 deaths are there. And the question is whether the corticosteroid therapy is going to reduce the neonatal death or not by observing these findings. I want your answer. You can type either yes or no. You can type either yes or no. By seeing those results, whether the corticosteroid therapy can reduce the preterm deaths or not. neonatal deaths or not? By seeing the data, the 2 into 2 table, I think you may get certain idea about how it will be. You can type yes or no in the chat box. I am unable to find your answers. Yes, very good. Preeti Sagar, no, no. Okay, very good. Nice answers. And like if we are going to generally observe that the proportion of the neonatal death in corticosteroid group is less compared to the non-corticosteroid group by just seeing the observation. There are 36 deaths are there, here 60 deaths are there. Okay. The thing is, after observation, we are getting that like there will be less deaths in the corticosteroid group. The statement is, I will say that it may happen due to chance. It may happen due to chance. It is not due to a significant difference between the groups. It is maybe happen due to the chance. Okay. Now the consideration is we are going to give regularly, routinely in any study, in any evidence. The thing is, what is the p-value, whether it is significant or not. This is the common terminology what we are going to use in our research practice. Okay. The thing is, depending upon the p-value, we can guess whether it is significant or not. The thing is, like if you can observe a scale, if the p-value is 0.5 means 0.5 means there will be the chance the result occurred due to the chance is 50%, and the result occurred due to certain, definitely it is there means 50%. And coming to the p-value 0.1 means the result occurred due to chance is 10%, and 90% the certain means it is existing in that evidence. It is existing in the result. Definitely the difference is there. And the p-value is 0.05 means 5% due to chance, 95% we can say that it is a true effect of the drug. This is the way how we are going to interpret the result. Okay, hope you understand the how to interpret the value. Generally, in any healthcare research studies, the minimum allowance for the chance is 5%. If you want more accuracy, means we can get that into 0.01 also. Okay, minimum allowable error is 0.05. 5% is allowable. It is very difficult to get 100% confidently. We can say that the result is having a significant difference. Okay, and like you can observe this table. Whatever the p-value, like if you are going to see the number in treatment arm 5, 10, 15, 20, 100, and the responses in the treatment arm 4 had responded. The proportion of response is same in the all the group. Okay, similarly, the proportion of the response in the control group also same in all arms. Like it is 0. In case of treatment group and 0.4 in the control group. If you are going to observe the p-value, like in 5 number of the patients, you got a p-value of 0.29, whereas in 100 number, you got a p-value of less than 0.0001. What it is going to indicate means, whatever the p-value, if you are going to increase your sample size, it will show at one point of time, it will show a significant difference. As a practitioner, you should not just blindly take the result based upon the p-value, whether that effect is clinically significant or not. Whether that effect is clinically significant or not. As a practitioner, we need to observe that. That is very important. That's the statement what I want to make from this slide. Okay. Okay, the same thing. The take-home message is not only p-value, whether that existing effectiveness or existing difference between the groups, whether it is clinically significant or not, you want to check that. Okay, then only we can make that that intervention into our practice. Right. The next thing is next parameter in the external validity is whether the patients who undergone in that research, those patients are relevant for our patient settings or not. It may be patient or settings what they had used. Everything we need to consider. Everything we need to consider. Why? Because the similarity, if there will be more similar, we can adopt that intervention. If there is huge differences there between the settings and patient population and their characteristics, then automatically it is not going to fit for our practice. Okay. After appraisal, then the fourth step is, sorry, the number is different. Like the fourth step is implementation of the evidence, and once everything is clear, means you are going to implement that evidence into the practice, and we are going to follow the intervention. It may be a short term or long term, and we will see that outcome whether it has been improved or not in the hospital setting. This is the way how we are going to bring evidence into the clinical practice. And now the point is, the objective of evidence-based medicine is to make a right decision by using high level of the evidence. And whereas even the objective of the artificial intelligence is also same, like to make the right clinical decision making by predictions given by the artificial intelligence. Both are same, the objective is same. Like just I want to give a brief basics of the artificial intelligence, and we will see how this artificial intelligence is going to solve the problems which are not solved by the EBM process. Okay, I'm not saying that artificial is going to completely replace the EBM process, but definitely we can say that it is a extension of the evidence-based medicine. Okay, right? Like if we are going to see the terminology, what we are going to routinely use in artificial intelligence means, one is artificial intelligence, machine learning, deep learning, data science. These are the terms which are going to used interchangeably. Like in the area of artificial intelligence, we are going to use machine learning. In the area of machine learning, we are going to use deep learning. Like that, interchangeably we are going to use, but actually it is not the right way. These are not synonyms. They are having their own specific function, right? Coming to the Venn diagram, in case of artificial intelligence, the primary objective of the artificial intelligence is to enable the machine to learn or think and make the decision based upon the whatever the data is available. There is a primary motto for the artificial intelligence. Okay, and whereas if you will come to the machine learning, it is a subset of the artificial intelligence where we are going to give data and we are going to observe the data analytics. Okay, we can say that machine learning means it is a data science, like it is a statistical tool which is going to explore and analyze the data. Like if we are going to see the machine learning, there are three types of machine learning is there. Hope you may know that like supervised machine learning, unsupervised machine learning, and reinforcement machine learning. Or reinforcement may also called as a semi supervised machine learning. In case of supervised machine learning, what we are going to do? We are going to give the labeled data to the machine. We are going to give the labeled data. Means, for example, like past data is available on height and weight. Okay, we are going to give the data like this is the input is height, and we are going to mention even output also. The for this height, the person is having this weight. Like that, we are going to give the labeled data to the machine, and the machine is going to learn the things. And once if you are going to give any new variable, it is going to give automatically what is the output. That is what we are going to call it as supervised learning. Everything you are going to give, that what we are going to call is a labeled data. Okay, in case of unsupervised learning, here we are not going to give labeled data, just we are going to give the data. The data may have wide variation in the characteristics, but you are going to give input for the machine to learn. A huge amount of data will be given for the mission, and the mission what it is going to do means it will based upon the similarity index, it will cluster the data. Is based upon the similarity index, it will cluster the data, and then each cluster there will be inter and intra comparison of the data is going to be made. And whenever you are going to give any new variable, the machine get predict the answer and it will give the decision. That is what we are going to call as a machine learning. Okay. In case of reinforcement learning, where the data is some amount of data is labeled data and some amount of data is unlabeled data. Okay, the machine is going to take time to learn the labeled data. Later, it will go through the unlabeled data, and then it will process the data, and finally, whatever the input we are going to give, it will predict the value based upon the so many statistical tools are there. I'm not going to touch all those things. And the last one is deep learning. Okay, deep learning is a subset of the machine learning, which is a subset of the artificial intelligence. Okay, we can say that deep learning is a core area in the artificial intelligence where there will be architecture of the multi-neuronal network. Okay, generally, there are most commonly used neuronal networks are artificial neuronal network and conventional neuronal network. Okay, and recurrent neuronal network. Okay, like in our brain, how the neurons and synapse, how they are going to architect them. Similarly, even in deep learning also, different multiple neuronal networks are going to exist depending upon our aim, how what type of artificial intelligence we want to generate. Based upon that, we need to select that program. Okay, generally artificial neuronal network, it is helpful to read the numerical data. Okay, and whereas if we are going to see the conventional neuronal network, it is going to capture the data from images. It is going to capture the data from the images. Whereas if we are going to see the recurrent neuronal network, it is going to observe the data in time series. There will be continuous flow will be there. If any new thing will be happened, it is going to detect it as a signal. Okay, the deep learning is
Narayan, sir. I think the voice is not audible, sir. You are in mute.
okay, sir.
Yes, sir.
Okay, sir. Okay, sir.
Okay.
Where it got muted, sir?
This slide, sir. Introduction of the slide. The current slide, sir.
Starting or ending, sir?
49, sir. 49. Slide number
49.
40.
Okay, here only. Okay, good, good.
Yes.
Okay, sir. Thank you. Right. Right. We had seen the complete basics of the what is mean by artificial intelligence, machine learning, and deep learning. We had a long discussion about what are the steps involved in the EBM process and to select the best research evidence and how to take the decision. Okay. In case of artificial intelligence, the tracking of the evidence, appraisal of the evidence, it is not going to be present. We are going to use the data. The data may be structured or unstructured data. We are going to give input of the data. The machine is going to learn the data. Slowly, it will generate the report, which is specific for the patient. A personalized report it is going to generate. Like if you are going to observe on the screen, there is clinical health data or personal habits data. So much amount of data we are going to give to the machine. It is going to learn. It is going to learn, and finally it is going to personalize the decision. Okay, that is the one of the major advantage of the going for the artificial intelligence. Okay, right. And as we know that, like majority, like all European medical societies, they are going to update their clinical guidelines periodically. Like day to day, the new evidence will come and they are going to update. Like if you are going to see the clinical guideline, we will be having two columns. One is for strength of recommendation, like strongly recommended, moderately recommended, or poor recommended. Another column is for the level of evidence, 1A, 1B, 1C, like that it will be there. Okay, the evidence is periodically, the clinical guidelines are periodically we are updating, and even this updation, whatever the updation they are making, it is not only from the RCT, even the evidence is from observational study, or sometimes expert opinions are also going to be included in the clinical guidelines, which we are going to follow those guidelines in the management of the patient. It is very excellent. It is required. Updation of the clinical guidelines is very important. Even today also, there are certain clinical situations are there where the evidence, whatever the available evidence, is not sufficient to address that problem. For example, if you are going to see the usage of anti-thrombotic medications in acute coronary syndrome or post-percutaneous coronary interventions or atrial fibrillation, in this, the evidence is there about use of antithrombotic medications in all these conditions, but in combination with chronic kidney disease, there is no one randomized control trial is there. Like for example, if any G5D stage of chronic kidney disease patient, if he is suffering with myocardial infarction or acute coronary syndrome, the choices, whatever the choice or drugs we are having, they are not from the highly evident design like randomized control trial. Whatever the evidence is available, it is even still we are depending upon only expert opinion or from observational studies. Okay, it is going to indicate that where the area where we need to implement the artificial intelligence to get a right decision. Okay, in this is the area where we can introduce the artificial intelligence, the data of the patients who are suffering with CKD and acute coronary syndrome, and what are the scores, bleeding scores are there. By using the multiple algorithms, we can get the scores and what is the risk of in each patient. We can individualize the outcome. Okay, This is the area we can introduce that. And a few more examples are there where artificial intelligence had shown positive outcome. A recent study using artificial intelligence, especially we can identify the patients who are having early risk for the chronic kidney disease, G1 and G2 stages. But in routine clinical monitoring or whatever the laboratory tests which are available, they are unable to predict the G1, G2 stage of the CKD. Okay, by using the artificial intelligence, Bayesian network and support vector missions, it was stated that we can able to detect the G1 and G2 stage of the CKD, so that we can take early interventions to solve the problem. This is a wonderful example. And one more example is about kidney transplantation. Okay, by using the artificial intelligence predictive models, we can predict the risk of graft failure or graft rejection. How much probability will be there in to fail that transplantation or graft rejection is going to be happen. Okay, almost 48 variables they had used in this predictive model, both from the transplant donor and as well as from the recipients. Okay, it can able to predict the transplantation rejection within first year, how much will be there, and within 3 years how much probability will be there to get that rejection. This is a wonderful example which are not addressed by the regular evidence, which is what we are following from the EBM. Okay, and you can observe that the left side is how the EBM is going to work, and the right side is how we are going to individualize the evidence by using the artificial intelligence. Okay, right. And finally, I conclude that the same statement, like artificial intelligence is not going to replace the EBM practice, but we can say that it is an extension of the EBM in clinical decision making process. Okay, finally, even if current guidelines do not include implementation of the deep learning program or machine learning program in the decision making process, definitely in future years the revolution will come where the artificial intelligence is going to play a significant role in the clinical decision making process. Okay, thank you very much. Thank you all. And I thank to organizing committee for giving this opportunity to deliver session on EBM and how the relation of the machine learning in relation to the EBM. Thank you very much. Any queries, questions? You may please. Due to lack of time, I am unable to show the databases also, how to search and everything. I think you are well equipped in that area. Thank you very much. Sahiti.
Yes, sir. Thank you, sir. Thank you.
Thank you.
I request the audience, if you're having any doubts, please unmute yourself. I request the audience to unmute yourself and you can interact with our speakers.
Okay, Ma Sahiti, I think there are no queries or questions from the audience side.
Yes.
Any other things, Ma? Sahiti?
No, sir. I think there are no questions from the question and answers box also.
Okay, Ma. Thank you.
There was a question, sir. 1 second.
Yes, Ma.
Sir, there was a question from Preeti Man. She's asking how artificial intelligence will help decision making about the selection of drugs and ratios of the drugs.
Yes, it is a wonderful question. Actually, Artificial intelligence is not like a conventional method how we are going to follow from the research evidence. Here we are going to completely depends upon the data, like electronic health record data or claims data or whatever the records, electronic records we are going to maintain in the hospital. That data, it may be either structured form or unstructured format. We are going to input the to the machine. It is going to learn, and it will predict the value of the outcome, whether that intervention is going to be having probability of success for that outcome. It is going to judge. That's the primary principle involved in the decision making from the artificial intelligence.
Yes, sir. Thank you, sir.
Yes.
There is a question from Prakash, sir.
Yes, yes.
Artificial intelligence will help in drug disease conditions.
Yeah,
Any
surely it will
AI
help.
tool? So the question is, any
Yes,
AI
yes.
tool is there to assess medication adherence?
I think up to now, from available evidences, there are no any artificial modeling for the adherence. There are certain studies in the area of drug safety and drug discovery and EBM decision making. Whatever I crossed from the literature, I didn't find any artificial intelligence work in the area of the drug adherence. But whatever we are going to use, what we will say that the container opening where we are going to keep a chip, and we are going to predict that how many times the lid of the container will be opened. This is the one solution of the artificial intelligence where we can predict the medication adherence. Okay, wherever the technology will be there, definitely there will be artificial intelligence will be there. But right away, to improve the medication adherence, directly there is no any model or technique in direct relation to the medication adherence.
Participant, you can share your questions in the chat box, or you can unmute yourself and you can ask the questions, please.
Preeti madam, you can unmute yourself and you can ask the question.
Sir, good afternoon, sir.
Good afternoon, madam. Please.
Sir, I think you may remember me because I have studied B.Pharm from our college, Preeti
Okay,
Sagar,
okay.
8 to 12
Yes,
batch.
yes, ma'am. Yes,
Yeah.
ma'am.
Sir, I am doing my research, research scholar I am. So I am
Okay.
trying to get the help of artificial intelligence from I wanted to check the ratios which are giving
Ratio.
the dosage of the drug which are giving the effect so I wanted to perform the nanoparticles, so I wanted to decide the ratios of the dosage. So how this artificial intelligence is helpful in deciding the ratios? I heard that they will artificial intelligence will give a triangle from where we can get the ratios. So I just wanted to know about this more, so that's why I wanted to talk to you personally so that
Okay,
I
okay.
can.
Dosages means again it depends upon the probability. Like there are certain Bayesian models and some models are there, madam. Like we are going to apply that model and we can predict the dose which is going to be accurate. Same statistical part only. Like till now we don't have direct deep learning level of the evidence, but we are going to use the statistical modeling techniques to predict the doses or outcomes.
Sir, is there any tool for the statistical analysis?
tool means normal like regression model or Bayesian model. Bayesian model is the right model which we are going to use to predict the doses or outcomes.
Sir, can you please come again?
Bayesian approach. It is a old technique only in statistics. Bayesian approach. Bayesian approach.
Okay, sir. Thank you, sir.
Thank you. Thank you.
Thank you, sir.
Thank you.
I hope the questions have been answered. So thank you so much, sir. Thank you so much for joining.
Thank you all participants. Thank you the organizing committee, AICTE and Triper. Thank you very much.
Thank you, sir.
Thank you.
So with this, we have completed our morning session. In the afternoon, we are having a session by Manasa. You can start joining by 2:30 p.m. Start by 2:30. I request the participants to join before 5 minutes. With this, we are going to conclude the session. The joining links have been forwarded to WhatsApp and the mail IDs. So please join through the respective joining link for the afternoon session. The session starts by 2:30 p.m. Thank you everyone for joining with us.
Um, Hello, sir. Good afternoon. So, is Dr. Mohanraj Garu is available?
Okay. Yeah, madam. Good afternoon.
Yeah, good
So I think
afternoon.
madam have joined it since. Okay. Yeah.
Okay.
Yeah, madam. Good afternoon.
Yeah, good afternoon, sir.
Yeah.
So, shall I start sharing my screen or start it?
Well, 2 minutes, madam. Remaining will be joining
Yeah, sure.
participants. Why? Because the previous session completed 1 hour back. We had two
All right.
sessions continuously. So
Okay.
what? A few minutes, madam. 5 minutes, please.
Yeah, no problem, sir. Yeah,
Yeah.
we'll wait.
Sahiti,
Sir.
could we start?
Um, yes, sir.
Okay. So you can invite Madam.
Yes,
So
sir.
she's ready to present.
Yes, sir.
Yeah, fine.
Good afternoon, all the faculty members, speaker, and participants. Welcome to the afternoon session of day 2 AICT ATAL sponsored online faculty development on drug safety monitoring and transition of care, the brink of an artificial intelligence, organized by Raghavendra Institute of Pharmaceutical Education and Research Autonomous. So for our afternoon session, we are having our speaker, Dr. Kondamadugu, Project Coordinator, Healthcare Intelligence Research Center, International Institute of Information, Telangana, India. Madam has completed her graduation B.Pharm Institute of Pharmaceutical Sciences, Siddipet, Mania University. Her post-graduation postbaccalaureate PharmD from Bharat Institute of Technology School of Pharmacy, JNTU, Hyderabad. After her post-graduation, Madam has joined as clinical research coordinator, Krishna Institute of Medical Sciences, Hyderabad, and the past work experience Drug Safety Associate, Quintiles Pharmacovigilance, Bengaluru, and Associate Professor, HOD, Clinical Pharmacy, Institute of Technology and Sciences, College of Pharmacy. And currently, Madam is working as project coordinator at Health Intelligence at iHub Data, IIIT Hyderabad, working with a team of eminent professors and research fellows on an initiative on leveraging artificial intelligence in the general area of healthcare. And Madam has done her internship in cardiology, Mahavir Hospital of Research Center, Hyderabad. And Madam has presented various papers during various sessions such as in 2010 organized by and has participated in APTI National. With this brief introduction, I'd like to invite Manasa Madam to start.
Thank you, Sahiti. Thank you for the warm welcome and having me here today. And I'm very much delighted to be part of ATAL FDP program as a speaker. And I thank Dr. Padmanabh Reddy Garu and Dr. Mohan Raj Garu for this wonderful opportunity. And so, good afternoon to all the dignitaries here and all the participants of this program. And I'm very much excited about the theme that is AICTE training and learning program, that is drug safety monitoring and transition care, the brink of an AI. And today I chose to speak to you all on facilitating artificial intelligence and drug safety. Okay, so let's not waste much of time and quickly dive into the topic. Let me pull out my presentation. I hope I'm audible.
Yes, ma'am.
Is my screen visible to everyone?
Yes, ma'am.
Thank you. So if you have any doubts or queries in between, you just raise your digital hands and I'll try to address them. And let's also keep the session interactive as there's a post-lunch session. And yeah, let's get started. So today I'm going to walk you through the pharmacovigilance and drug safety and revise some definitions and learn about the case processing workflow in pharmacovigilance and what is artificial intelligence and its subsets, applications of artificial intelligence in pharmacovigilance and its benefits, and the future of pharmacovigilance. So what is an adverse event and an adverse reaction? I hope you all must be knowing about this, but I just want to give a quick revise on this. So any medical occurrence temporarily associated with the use of a medicinal product, but not necessarily causally related, is an adverse event. So, and sorry to stop in between. I just would like to understand if there is any technical glitch while I'm speaking, because I mean when Saiti was speaking, I couldn't hear properly. So I just want to know if it is clear.
No, madam, it is clear.
Yeah, thank you. So, okay, so what's an adverse reaction? It is a response to a drug which is noxious and unintended and occurs at doses normally we use in human for the prophylaxis, diagnosis, or therapy of any disease, or for the modification of the physiological function. So these are the quick definitions of this adverse drug reaction and event. Let's move on to the pharmacovigilance and drug safety. So what it is? So it is the science and activities related to the detection, assessment, understanding, and prevention of the adverse effects or any drug-related problems. So to understand this definition, I think we need to first understand what is benefit and risk ratio. So all the drugs, along with some potential beneficial effects, also have some side effects as well. However, the benefit of the drug is more than its side effect, but sometimes the risk may be higher than the benefits of the drug. This is like to evaluate this. This is the benefit and risk ratio. So let us now come to the first step of the pharmacovigilance, that is detection. In this step, we detect any new signal of the drug. New signal means any new safety information about drug use, and which affects the benefit and risk ratio associated with the use of drug. In simple language, we say that the benefit of the drug is less than the risk associated with it. So, okay, there's a quick question. Does anyone know the significance or importance of today? Maybe if someone can just quickly. Okay, so I just would like to take an example of a drug. So today we celebrate or observe today as a World Hepatitis Day. This is observed, any clue, who has discovered the hepatitis B virus? Okay, I hope I'm audible.
Yes, ma'am, you're audible.
Yeah, so quickly, can anyone? All right, so no problem. So today it's a birthday of Dr. Baruch Bloomberg, who has discovered hepatitis B virus, and we observe annually on 28th July as a World Hepatitis Day. So with that, I take an example of a nimesulide drug. And this is an analgesic which is usually given for headache and fever. Okay, so however, it has a severe side effect like liver damage when taken in a longer run. So here we detect the signal of nimesulide as liver damage. The next step is assessment. So in this step, we assess the severity of the detected signal. For example, in a previous example of nimesulide causing liver damage, we need to assess if liver damage is a serious issue arising due to the use of nimesulide or not. So for better assessment of signal, we need to collect enough data on signal. Okay. So the next step is understanding the adverse event. And in this step, we understand the signal in detail based on various nimesulide parameters like severity of the signal, benefit and risk ratio of the signal. Okay. Again, if we take the example of nimesulide drug, to understand this, we understand that majority of the people who take this nimesulide on longer run needed liver transplants, and many of them have died. So hence we can conclude that benefit of the nimesulide is less than its side effects. So ultimately, the last step is prevention. Let's directly jump into the example. Based on all parameters, the drug nimesulide was found unsafe to use, and it was banned from the market considering the patient safety. So this is what exactly the pharmacovigilance is. So here are some quick services of pharmacovigilance. From the solicited sources, the data is collected, recorded, and notified, and then data is entered in the safety database. Then the case processing workflow takes place based on the signal detected, and then the regulatory authorities are responsible for detecting and managing these safety signals, and insightful data analysis is done by monitoring the literature and social media like Facebook, Twitter, and any other data sources screening. So here is the case processing workflow. Once we receive any adverse event report from any solicited sources like the study reports or some clinical project reviews or clinical investigation brochures, case reports, we need to database these reports in the safety database. The day on which the company who is manufacturer of this drug receive any adverse event report, that day is called as day 0. So am I going a bit fast, or do you want my pace to be a little slow? Okay. So we have.
It's okay. You can move in this way.
Yeah, sure, sir. Thank you. So okay. So we now understand what is day 0, right? The company, the day on which the company who is the manufacturer of the drug, who receives any adverse event report, that day is called as day 0. Reports in pharmacovigilance is also called as individual case safety reports. All right. And day 0 is referred as regulatory clock start date. To understand the steps in the case processing, we need to first understand the regulatory timelines. So the 7-day timeline is for fatal and life-threatening, a 15-day timeline for other serious cases like cases with seriousness of hospitalization, disability, etc., and 90 days for the non-serious cases. So this is the time frame that manufacturer of the drug need to provide detail of report to the regulatory authority, and therefore we need to prioritize the cases as per their seriousness. So the process starts with confirmation of valid reports. Once the reports is valid, you need to search for duplicate reports in the safety database. Then the most commonly used safety database in the pharmacovigilance are Argus, RSG, RAVE, etc. So the first step in case processing is triage of individual case safety reports. If case is valid with all 4 validity parameters, then the triage associate can database the report in safety database and enter all 4 valid criteria. Once it is done, the associate can ask a query on missing information, if any, and can push the case ahead to the next workflow step. So this is the second step in case processing, that is data entry of individual case safety reports. The data entry associate will now enter the data in safety database. The usual responsibilities of data entry associate includes accurate and complete data entry into the safety database as per the report, and proper coding of the medical terms as per the report. That includes some medical history and other events of the patient through medical dictionary for regulatory activities, that is MEDRA, and then paper coding of the drugs as per the report with the help of WHO drug dictionary for the case. And the associate writes a narrative for the case and asks query if any, and pushes the case to the next workflow. And the next step is the quality review of the individual case safety report. In this step, the associate will thoroughly check quality of all the data entered by data entry associate, and he will also check whether the data entered is complete and accurate. Proper coding of the drugs and medical terms with appropriate dictionary was performed or not. Also checks the narrative, correct the cases as and when needed, and gives a green flag to all the data entered by the data entry associate and asks queries if and pushes to the next workflow step. That is the medical review of this individual case safety report. Here, the medical reviewer is a doctor by profession, and his or her responsibility is to check the causality of the reported event with the drug, seriousness of the event, coding of the medical terms, and can write company comment. The reviewer also checks the case for medical sense and asks for query if any. So the last step is submission of individual case reports. Submission associate checks the case for completeness of the case, then will perform the validity check of the case, and then submit the case to appropriate regulatory authorities. So this is the complete cycle of the case processing. Just to summarize the steps involved in case processing, it just starts with the triage or data entry of the case, quality review of the individual case safety report and medical review, and then finally submission of these safety reports to the appropriate regulatory authority. Okay, so the entire process of recording and evaluating adverse drug reaction data of a drug is one of the crucial steps in the pharmacovigilance and is defined in numerous guidelines to various degrees, and it is tracked by the regulatory authorities and drug marketing stakeholders. So let's move into artificial intelligence. So it is defined as a program that exhibits cognitive ability similar to that of a human being. So it is the ability of machines to learn and solve problems. In short, AI is next generation technology which can replace manual activity to automatic business processes without any human intervention. Okay, so what is the mechanism of artificial intelligence? So this artificial intelligence is a computer algorithms to improve automatically through experience. For this, AI is used as a machine learning. As per John McCarthy, one of the pioneers of the artificial intelligence, AI is the science and engineering of making intelligent machines. So to understand this better, we'll quickly learn about what is machine learning. So machine learning is defined as a study of computer programs that leverage algorithms and statistical models to learn through inference and patterns without being explicitly programmed. So this machine learning quickly evaluates the data and try to find the pattern. In the machine learning data, the desired result will be included based on the computer self-develops an algorithm. Okay, so the principles of machine learning is logic based on specific factors that are input in the computer, and based on that factors, computers try to identify the general principles which is used in the discovery of new medicines. So here we have three types of machine learning, that is supervised learning, unsupervised learning, and reinforcement learning. So in supervised learning, the machine learning algorithm is trained on labeled data. In unsupervised learning, the machine learning algorithm is trained on unlabeled data. I hope you understand what is labeled data and unlabeled data. So this reinforcement learning directly takes inspiration from how human beings learn from data in their lives. So it features an algorithm that improves upon itself and learns from new situations using a trial and error method. So favorable outputs are encouraged or reinforced with a reward system, and non-favorable outputs are discouraged or punished. So based on this machine learning continues. So machine learning continues to be applied in various fields and use cases. Like these algorithms always help to improve user experience, and also for customization of some online platforms like Netflix, Facebook, Google, Amazon, to provide unique content to individual users. And based on their likes and dislikes. Maybe, if you open your Netflix and you watch some web series and movies, based on that, when you turn it off and turn it on the next time, you'll get some recommendations. So this machine learning is working in the background to give you such kind of recommendations based on your likes and dislikes. So moving to deep learning. So what is deep learning? It is a subset of machine learning. And this principle follows the methodology of neural network of human brain, just like how our human brain performs. The same way, the deep learning technique also works on the same principle. So deep learning is a machine learning technique that constructs artificial neural networks to mimic the structure and function of the human brain. All right. So we have, if you look into this picture, the one on the right side, that is deep learning architecture. Here you find a input layer and an output layer. So you give some information and it will assume that the input data is a matrix of pixels. Okay, then the first layer typically abstracts the pixels and recognizes the edges of features in the image. The next layer might build simple features from the edges. Consider we have given an input of an image that is image of a tree broken into small pixels. It will, the first layer will build simple features from the edges, like such as leaves and branches, and then the next layer could then recognize it as a tree and so on. So the data passing from one layer to the next layer is considered as transformation, and turning the output of one layer into the output for the next layer. So each layer corresponds with a different level of representation, and the machine can learn with features of the data to place in which layer or level on its own. So here, this deep learning architecture, it is actually in case of pharmacovigilance. Okay, we'll come back to that. So there's let's move to the next slide. Okay, so here in this figure, as you see here, artificial intelligence for drug toxicity and safety. So we check in both preclinical toxicity and post-market safety. So here the AI methods that are used in our for preclinical toxicity, we use machine learning, and for the post-market safety, it is deep learning. So in the preclinical toxicity, machine learning techniques such as logistic regression and random forest are used to determine the interpretable models and the low orders of complexity, which are again used to predict drug toxicity, and it aids in trial design and drug development. Whereas in post-market safety, the deep learning techniques that are used are convolutional and recurrent neural networks. Here, these are highly nonlinear models, and they have higher orders of complexity. These are used in monitoring real-world use and comparative safety analysis. It also aids in clinical decision making and in some regulatory actions as well. So moving back to the previous slide. So here the applications for pharmacovigilance. So the good news is that nearly every process in drug safety and pharmacovigilance can be automated. And automation supports operations right from the data entry, that is triage, to the report submission. Like in all the steps, like quality review, medical review, and finally the submission of the individual case safety report. So automation coupled with artificial intelligence improves the quality of safety data at the start of the process and helps streamline processing, and it also improves signal detection and evaluation. If we let artificial intelligence perform this highly repetitive manual labor of extracting and reading incoming reports, it would free up time for pharmacovigilance experts to review and revise the reports. And machine learning clustering models are used for detection assessment. With the help of deep learning, thus pharma companies can reduce the time taken to report adverse drug reactions. NLP, that is natural language processing solutions, help in processing unstructured and free text form into standardized digital version and normalized case reporting. Additional data sources such as patient records can be structured by applying this natural language processing to support signal detection and verification. Speech and text recognition further enables companies to deliver error-free reporting with natural language processing. And we can also mine literature and unconventional data sources. The techniques emphasize predictive accuracy over model interpretability. The predictive analytics can also be used to evaluate insights and potential signals in the data that were very difficult or impossible to identify before. So these deep learning powered language processing methods, including word embedding and attention mechanisms, are the techniques of choice to extract adverse drug events and are related to this text data. So are my slides visible? There's some technical glitch.
No, slides are visible.
Thank you. Thank you. So coming to the benefits of artificial intelligence in pharmacovigilance. So as you see, it is very cost-effective process, and it enhances in expedited reporting. It lets pharmacovigilance experts do high-value work, whereas this can actually give insights on safety reports and just a minute. It ultimately it also gives some good regulatory compliance and enhances the patient experience and the patient safety. So the future of pharmacovigilance. This artificial intelligence, this holds a great promise for safety and pharmacovigilance, and is already proven in the industry. Automation, artificial intelligence, and machine learning technologies provide opportunities to shift the pharmacovigilance function from compiling and reporting data to help increase product quality, optimizing treatment plans, reduce costs, and improve patient safety. And overall, it increases the drug safety. It lies, I mean, the future of pharmacovigilance, it lies within the digitalization of the data, artificial intelligence analytics, and patient-centered data collection, which will likely increase the overall drug safety. So sometime when in future, when the machine learning crosses the human intelligence, this point is called the singularity, and later it would be termed as artificial superintelligence. So these are some quotes, okay, by Stephen Hawking, Bill Gates, and Elon Musk. Few believe this is helpful, and few believe that this can destroy entire humanity. However, this is just an hypothetical situation. When it comes to pharmacovigilance, the overall risk of implementing AI approaches is low, and the opportunities are very high, as it may have a positive impact on healthcare and reduce costs. So with that, I conclude and would like to take up queries, if any. Thank you.
Sahiti,
Thank you, madam.
open the
Yes, sir.
section for question and answer.
Yes, sir. Thank you, ma'am. I
Yeah,
request
thank
the
you.
participants to unmute your mic and interact with the madam. If you're having any doubt, you can post in the chat box having question and answer session, or you can unmute yourself and you can ask question.
So, may I ask a doubt regarding?
Yes, please.
Yeah. So, means whatever you have discussed is from a practical point of view
Yes,
regarding the particular job nature or a job description. But right now,
yes,
in India, AI has started to rise somewhat in a good way, where we
yes,
have tied up all the processing requirement with the USA.
yes,
So I think recently I came over a article on comparing the economic countries and putting AI in Indian aspect at the crossroad. Like that article I have came across, where nearly maybe 3 to 8% of the GDP right now is spent on AI by the
yes,
government of India, which is nearly 3 times the fold in China. So how about the development will be there? In what aspect the healthcare will be able to take up this recent methods of AI into the healthcare, and how safe it will be delivered to a patient, and making them aware about AI. So right now all the technical peoples are speaking about AI. Okay, but the main objective of AI in healthcare is to make it reachable to every consumer, and that consumer must be made aware about it. So whether India is working on it or else the MNCs are working on this.
yeah. Okay. Thank you, Dr. Mohan Raj Garu. That's a wonderful question. So, yes, in India, our government is taking many measures to bring awareness in AI. Of course, I mean, this all depends upon the huge data that comes up from hospitals and from doctors, individual doctors, and the patient data is it's very critical in certain cases that it needs anonymization. But then uh, government of India is actually taking many such precautions in the safe use of data. And when coming to the awareness, yes, I mean, at large, when these techniques are implemented and the tools are developed in healthcare, AI tools that are developed in healthcare, yes, they are doing a lot of dry runs. They are doing a lot of dry runs, and they are having the interacting with doctors, patients, and yeah, many such. I mean, we actually don't know, but behind the screen, yes, everything is actually happening. And since past 2 years, as I'm into this, I see there are many areas across pan India. People come up with good problem statements or use cases, and we actually collaborate with different institutions and hospitals across India, and we are trying to uh, gather this Indian cohort data sets for for individual, I mean, the different kinds of diseases. So we actually, I mean, before collecting these data sets, particularly for these kind of development of AI tools, we bring across this awareness among all the patients, because ultimately we need to take that patient consent, and there are many such ethical procedures, as you all must be knowing. So yeah, everything is going on in full swing, and maybe in a couple of years we will actually come across many. I mean, there are some tools already which doctors are using in detection of breast cancer using this mammography images. And we are also trying to develop some or curate some our own data sets for Indian cohorts at IIITH. Yes. I hope this makes sense.
Yeah. Okay. Good. There is a question. Kathy, you can read the question from the chat box.
Okay.
Yes, sir.
I'll just go through.
Madam, there was a question from Patil, sir. So, sir is asking to know about how a software detects signal, whether it is from a patient's description or from the investigator's observation. So, how the artificial intelligence or the software will detect the signal?
Yes. So, sir, that's a good question. So, here we have some AI tools like prescription screening, wherein like you can see in India. I don't think we have still come up with such solution, but we have such tools which can actually screen the prescriptions and detects the drug-drug interactions and adverse drug reactions. So here, based on the prescription, okay, which a doctor writes, and the medicines that are present in the prescription, we give that input. Say, suppose we are using deep learning technique in this instance. So we give input as these input as drugs, and we observe. I mean, there are some hidden layers inside this architecture, wherein each and everything they will correlate the prescription of drugs with one another to analyze the drug interactions or any adverse drug event. Suppose if there is an adverse drug event, based upon the chief complaints of the patient, that is given as an input, and in that way, it is like both patient centric and doctor centric, and ultimately with the inputs that we give, the output, we detect that output. And also natural language processing is also plays a key role here in detecting or identifying the text data.
Thank you, ma'am. So, any further questions? The participants are requested to unmute your mics and can ask with the madam. Directly can interact with the madam.
Hello, ma'am. Myself, Dr. Mahendra. I would like to ask this question: how well these neural networks and the latent variable models and also anomaly detection systems will work out with respect to pharmacovigilance is concerned in an AI? Because as I know, the k nearest neighborhood and the decision trees are not accurate all the time, and it's leading to many variable outputs. So how well this will be understood as far as the pharmacovigilance is concerned, and also how well we can resolve these systems?
Okay. So, okay. So you would like to, I mean, let me, I missed out on a point. Can you just repeat yourself, please?
Yes, I want to know how well the neural decision trees and the anomaly detection will be worked on with respect to AI in pharmacovigilance.
Okay. All right. So, uh, I'm sorry. I'm sorry. Maybe I am not the right person to answer this question because I haven't worked on this.
Madam, could you elaborate your job role? What exactly, how you are working and how you are managing project, and what is your I mean to say that how you, as being a family graduate, assure you that AI is going to be helpful for Indian healthcare.
Okay, the PharmD graduate into AI. Okay. My job role here at IIT Hyderabad, initially I joined as a project coordinator, wherein I were like a liaison between professors and research fellows and postdocs. The projects which they work, I mean, in with respect to healthcare, if they have any if they have any queries related to healthcare, I was like a one-stop solution for them. So in that way, slowly I started taking my first steps in AI. So initially I mean, this is what it is, and I slowly started learning Python and AIML techniques. But when coming to your second question, how AI will help PharmD graduates? Yes. If a PharmD graduate learns these AML techniques, I mean, keeping a vision in mind about the future, maybe it's like, nowadays, I mean, since past 2 years, what I'm observing is it's like a collaboration between medical professionals and computer scientists. Okay. So in future, if we as a pharmacy graduates take up this AML solution and AML techniques, maybe we can, I mean,
Excuse me, ma'am. Excuse me, ma'am. You have been muted.
I'm so sorry for that. I was explaining. Okay. So thank you. So I actually answered the first question while I was on mute. Okay. So
Madam.
Yes, sir.
Don't mind it. I'm not yet clear with your answer because see now as you are telling that a PharmD graduate need to study Python theory, right?
Yes, sir. Was I on mute?
No, indirectly. Hello, can you hear me?
Yes, but was I on mute while answering, or was it audible?
You are on mute actually.
Okay. Yeah, please go ahead.
So at the end of the day, I think because the topic which the Riper has taken is very important and essential topic right now.
Yes.
And now, as my question is, what I asked you is how you are like being you. Many family graduates may follow you in the AI also in the future. So you are being a speaker. You are taking a talk. So most of the faculties who are attending it are also thinking to tend to tell our students also to join AI. Probably the future is. So I want you what exactly you are doing it. So probably I can also suggest my students also to probably AI is also one of the options for you for your future.
Yes, sir. Absolutely. So we actually have to recommend our students to take this AIML courseworks. Okay, because in future, I mean, right now we are acting like a bridge between, AI tools. I mean, developing these AI tools, we are like a bridge between the medical professionals. I mean, it's like always, pharmacists are always like a bridge between, medical professionals and patients. But here, when coming to developing AI tool, we are again like a bridge between the medical professionals, computer scientists, as well as with this research fellows who are coming up with these solutions. So when we as pharma graduates, when we take up these courses, we might no longer be like a bridge, and we can actually take that as a coursework, and we can do our own projects, and we can directly interact with doctors. We may not require computer scientists or any engineers or something. We ourselves can do that.
Don't mind me in asking this, ma'am, because you have clearly said that pharmacy graduates can play a vital role in this bridge,
Yes.
right? But you're also asking us to concentrate on the other particle, like engineering models, right? It may be a
Yes.
linear kernel model, polynomial
Of
kernel
course.
model, and
Yes.
the radial basis canonical model. These are the models which you normally use it in AI,
Yes.
if I'm right. But you also concentrate like this includes typical and concentrated based theories, right?
Yes.
So, because I have also worked as a data developer for a AI-based company in Australia, which is located in
That's
Australia.
great. That's great.
But what you are saying is totally different than what we were doing it in there, right? We
But
normally,
sir, in future, it is definitely going to change the perspective.
I'm not talking about the perspective as a whole, but the job role which we played there, and it's very important we understand the job role which we played there. Exactly right. So you're saying that we act as what an engineering guy can do, but it's very important that we also concentrate on a patient-based centric studies, right?
Yes, yes.
Patient-based
That is
approach,
what, like when
wherein we understand the normal physiology-based things, not exactly what the AI based. You may be asking, there are many AI tools which are collecting the patient history forms, right? We have an applications wherein they collect the patient history of a patient. The examples, maybe Bangalore has started a new company, a startup based company,
yes.
right? But where we are failing is the accuracy and the precision tools, right? How well we can ensure that accuracy and the precision models which we are establishing through the AI models. Because as per my knowledge, because I have gone through some of the articles and some of the information which is available on the internet, because this what the data says is the precision tools are always leading to the wrong decisions sometimes, and it's always not possible to detect. Because the other day I was speaking to a doctor wherein he clearly said that these AI tools are replacing the doctors, yes, but at the same time it's also leading to wrong decisions. Now how will you justify this?
No, it doesn't replace doctors. First thing, it will just enhance their work, and it will ease their work. That is what my comment is. It doesn't replace anybody. Be it pathologist or radiologist, because we come up with image segmentation techniques. Okay, so it never replaces any doctor or pathologist or radiologist. It just enhances their work.
Sorry, ma'am, don't mind me in saying this. There was
Yes.
a report given by the international community on AI, which normally released by US company. Okay, there says that these AI tools are in the rural parts are replacing the doctors, but it's not giving the accurate information or the accurate precision with respect to diagnosis is concerned. You're saying that yes, enhancement is there with respect to data collection, but how well the data is stored and what's the perspective in the re-implementation of these data are to be our prime concerns, right?
Yes, data anonymization is a prime. I mean, data of a patient is a primary concern for in any in any perspective, but we actually follow certain ethical guidelines, right, to anonymize this data. So, and coming to those reports, I mean, I didn't go through that. Okay, the article which you have gone through, but at my workplace, we have data foundations wherein we anonymize all the data, and even we have medical professionals like doctors who actually are trying to implement these AI tools to in their medical camps. Okay, not in hospitals, but in medical camps. But then they never say that it is replacing their work role. Okay,
Sorry for this,
they
ma'am,
never say
because
that.
I have worked as a data developer and also I have
Okay,
worked in the very grassroots levels.
yes,
I
but
often speak. I often speak to many doctors while I promoting my product because
okay,
almost all the time they say that yes, this is good, but at the same time it's also have a pro and cons. Wherein the cons are more in the AI. As per the data collection is concerned and how well we segregate onto on the anomaly theories which we have, the linear kernel theories which we are using in the data
yeah,
collection and also for the implementation of that data into a software form or a PDF form, right?
okay.
I'm a little bit more concerned about how well this data will be stored because very recently, WikiLeaks has given a data how well these data patient data is used or misused for the other causes.
Mm-hmm. Yes, of course, I agree on that point. But at IIIT, we are actually taking all the precautions not to leak the data, or we are anonymizing the patient data. So, like as of now, we have never encountered the point that you have just brought up. But so far, it was going very smooth. And even, I mean, there are many collaborations happening around, but we have never heard such things from the doctors which we are working with. So I'm really sorry, because this is what it is at my end. But at your end, it must be different, because not everything, I mean, everything is same around. So yeah, I can't comment on that.
All right, thank
So,
At
madam, could
my
you
end, at
let
my
me
end,
know? Okay, yeah,
yes, please.
please. Yeah, please. You can continue, madam.
So I just want to say that I mean, we are taking initial steps in data foundations. We are gathering data, curating our own data sets, and we are coming up with some AI solutions with data driven technologies. So yes, it is. See, the concerns which sir has just told, I mean, that is really a concern, but it's not a same in all the cases. So at our end, we don't find anything as such, and the doctors or other medical professionals which we have collaborated with, they have never come up with such kind of I mean, they never told that they are replacing our work or our job role. I mean, it never happened so far, because it's been more than 1.5 years that we have started up with this healthcare initiative. Yeah.
So, from my side, there is one last question.
Yes, sir,
So,
please.
maybe AI will have a very good role in healthcare, but as per the previous participant, there are
Mhm.
many limitations around AI in healthcare. So, whether came across working with peoples in framing any action plan or
Yeah.
any type of strategy, where
Yeah.
all the AI projects cannot end with the positive responses. It may have certain limitations. So if so, how you develop an action plan for those limitations? The project you have came across, your experiences.
Okay. Okay. So far we have worked on, I mean, since past 1 year, we have worked on, say, roughly around 16 projects. Okay. So in this 16 projects, like each and every project is entirely different from one another. Right from drug discovery, or developing a tool for detecting this COVID-19, and detecting healthy aging of brain. So there are like many such kind of projects, but we didn't come across any such kind of limitations. I mean, all the works which we have done so far had no such limitations, or we didn't face any challenge so far. and we just began with a few projects recently. But I don't see any such limitations because whoever is collaborating with us so far, we had, everything is running smoothly as of now. We didn't had any limitations as such. Yeah.
It was nice. And could you give your recommendation how well the upcoming millennial graduating students, either from science, technology, or medicine, can prepare themselves to apt a job in artificial intelligence? How they have to prepare themselves?
The pharmacy, life sciences, or we're talking about which?
Science, technology, engineering, and medicine.
Okay. Sir, to be frank, we don't have any such courseworks in India which will actually aid our students to learn these AI/ML techniques. So I think there should be something like that should be incorporated in our in our curriculum, so that it will be helpful for our students, because this is like AI generation now. So I think there's an urge to incorporate such kind of curriculum, because there's an interdisciplinary thing for us. For us, especially for a life science background student. So it's always good to have knowledge around what is going, see, initially when I started at IIIT Hyderabad, I was like very new to all this terminology. But slowly I got acquainted with this, and I'm learning. I'm still learning. I don't say that I am good at AI/ML now, but I'm still learning. So if there is any particular curriculum that is framed, maybe that would be useful for our students. And I think there are many such initiatives that are going in and around. But I think, if AICTE will look into this and try to come up with such courseworks, that would be really grateful.
Yes, thank you, madam. Participants, if you want anything to be cleared on the practical aspect of PV and drug safety in relation to AI, kindly post your questions in the chat box, or you can unmute yourself and ask the questions to madam, please.
Or if you are looking for any courseworks, maybe I can share that with you.
Yeah, ma'am. Sure.
And also some study materials, so that you can share with all the participants.
Sure, madam. Sarthi?
Yes, sir.
Can you look into the chat box, ma'am, so we can...
Yes, sir. Sir, till now it doesn't have any questions.
Okay, Sarthi, you can give a thank note to madam.
Yes, sir.
Yeah.
Thank you, ma'am. Thank you for joining with us and sharing your experiences, how artificial intelligence will work out in pharmacovigilance and drug safety, and how the case processing will be there. Thank you for joining with us and sharing this knowledge. And we are very happy for us that we have.
Thank you, sir, for having me today.
Nice presentation.
Thank you.
Application-wise, it was good. Yes.
Thank you, sir. Thanks, everyone.
Okay.
Excuse me, ma'am.
Yes, please.
There's a question.
There was a question.
Okay.
But you're again doing the same.
Bye, madam. Thank you.
Whether the AI tools useful for design a project work at PG and PhD level. There is a question from Karthikeyan Ramadas.
So, Karthikeyan sir, can I have a direct talk with you? What exactly are you looking for?
Karthikeyan sir, you can unmute yourself and talk, sir. Karthikeyan sir.
So am I audible, sir?
You are audible, sir. You can directly interact with madam.
Yeah, okay, sir. So just I have a small question, sir. So by
yes,
using
sir, please go ahead.
by using artificial intelligence tools, whether we can design any project work for UG or PG or PhD level students, especially in pharmacy.
Yes, sir. There are many such projects that we can do, but again, it requires an expertise, AIML expertise. So we would definitely need to collaborate with some engineers to do that projects.
Okay. One more question, madam, if you don't mind.
Yes, sir, please. Yes, sir, please.
Is there any short-term courses are there to take up by our UG students or PG students?
Yes, sir. Definitely, I'll keep you posted. I'll share it with Dr. Mohanraj Garu, so that he'll share across with all the participants. Maybe that would be useful, because I definitely feel an urge that our students should take up some courses like this. I'll definitely share that.
Thank you, madam.
Thank you, sir.
Thank you.
Thank you, sir. Thank you, ma'am.
Thank you, ma'am. Thank you.
With this, we have concluded our day 2 session. The feedback link has been posted in the WhatsApp group and also in the chat box. I request the participants to complete the feedback procedure by tonight. And please ensure that you are entering the correct participant number. The participant ID has been given in the PDF document which has been circulated in the WhatsApp group. Please check the participant ID correctly and fill it correctly so that your attendance can be considered.
So, dear participant, the link will be active and functional up to 9 o'clock at the night, 9 p.m. So kindly provide your responses at the earliest. And we join for tomorrow. And tomorrow we have one first session by Dr. Chakka Gopinath sir on moral value and ethical living as part of a mandate session as per AICTE, and followed by 2 technical sessions handled by Professor of Technical Educations. Both are from Tirupati, and one person is the head of faculty of Vellore Institute of Technology. And so what I think you'll be getting into a very practical applications of machine learning and Internet of Things. So I think it will be very interesting for you. Kindly join morning, and the link of all the 3 sessions will be shared to you both in mail and also the group by late evening. All right, and you can join, please. And don't forget to give your response on the feedback, since it accounts for your attendance. All right, and give your
one more thing I want to add.
Yeah,
sir.
Please.
Yeah, dear participants, whenever you are filling feedback form, in the registration number, please don't enter your phone number. So even today morning we have said, our ma'am Saiti has said, but still much mistakes are there, because this is an automated thing and you may face difficulty in getting your certificates at the last. So please try to read properly the question which was given and enter your registration number properly. Please do that. Thank you.
Thank you, sir. So what you just participant, make sure that you are entering your ID number, and the particular PDF has been shared in the WhatsApp group. Please look into your name, your ID number, then start providing your response. Thank you. We'll meet tomorrow morning. And we end the meeting for
Thank you all for joining with us.
Yeah.
We'll be signing off. We'll meet here by tomorrow morning. Thank you all.